Thank you, Dr. Lee, Professor Lee. I just finished up your microphone. Okay. Thank you. All you. We have a few financial support from National Science Council, the solid one, Science Technology Council, and also from our Thank you. Sometimes after fun, we will share the
increase research performance and contribution to reduction physiology. Today, Dr. will give an overview of the Finnish reproductive physiology of the conservative physiology, physiological and evolutionary point of view. Thank you.
Sawan. It is my great pleasure and honor to be here today and to participate in the Chinese conference. In fact, after more than 30 years of collaboration with Professor Chong Qing-Hong, I feel like Taiwan and Keelung are my second home. So I'm very pleased also for that reason. So I give all my thanks to the organizing committee and Professor Chong Qing-Hong for having invited me to give you this introductory conference. I will talk to you today about some historical point of view of fish reproductive physiology, going from comparative to evolutionary and ecological physiology. I'm working at the Museum of Natural History, and this is the exhibition gallery for the public, the marine section of it. So I hope you will, the one that have among you that have not been yet to Paris, you will come and visit us one day and visit our exhibition room. The Museum of Natural History in Paris is an institution toward the public, but it's also a research institution and higher education institution. We have research laboratories, we have PhD program, and I'm appointed by CNRS, National Center of Scientific Research in France. And together with Sorbonne University, we have founded the Ocean Institute, which gathers scientists from various fields, from biology, ecology, but also from paleontology up to looking at the history, it is considered that the world of physiology was created by a French scientist, a French astronomer, mathematician, and physician, was a doctor of science. He wrote a book named La Medicina in Latin, and with three big chapters. One of them was physiology, the other pathology, and the other therapeutics. So it is considered that the word physiology was founded by these scientists in this period. And physiology is defined as the branch of biology that deals with the normal functions of living organisms and their parts. So at the beginning, it was dealing with human physiology in the frame of human medicine. We have technical problems. One big step was made 3 centuries later, again by a French scientist. His name is Claude Bernard. He was again a physician and physiologist, and he founded what is called experimental medicine, which is together experimental and comparative physiology. He set up what is experimental physiology, which the pattern of observing, making hypotheses, experimenting, raising results, interpreting the results, and raising conclusions. And this experimental approach is today still the one that all of us are following. And interestingly, this Claude Bernard professor was the professor at the laboratory we are still in at the Museum of Natural History. And he also initiated comparative physiology by investigating various animal models, choosing the right model for the right question. So it extended physiology from human questions to animal and comparative physiology. So that's an important milestone. So if we go now to fish physiologists, it's at the 20th century that we find some of the pioneer research scientists that set up this disciplinary field. In France, there was Maurice Fontaine. Again, he was one professor of our laboratory. And in USA, we had Aubrey Gorman at Washington University and Howard Byrne in Berkeley. Some of us have been lucky enough to know them. But what are fishes? If we look at the first classification of species based on phylogeny, it means classification based on evolutionary relationships between species. The first classification such based was performed by a German scientist, Ernst Haeckel, in this period. And he divided the living kingdom in 3 big parts: plants, protists, animalia. And fishes, like land fishes, are found in the animal kingdom and belonging to vertebrates, animalia. Nowadays, with all the advances in phylogeny, including molecular phylogeny, it looks like that, the phylogeny of the animals or metazoa. And vertebrates are here, part of chordates, deuterostomes, bilateria, metazoa. And what about fishes? So here, about fishes, we have the teleos. The teleos is the largest group of fishes, and it belongs together with some other groups, such as holosteans, in what is called actinopterygians. So that the largest number of fishes that are studied belong to this group of teleos and actinopterygians. But there are also other species named fishes, such like the cartilaginous fishes, sharks, ray, and they belong to a completely different group, which is more basal, and it's the chondroitians. And then there are also other species named classically fishes, which are the cyclostomes, like the lamprey. And again, they belong to a completely different groups, which is the cyclostome, and which represent the most basal groups among the vertebrates. And it's not all. There are some species, usually named fishes, that again belongs to a completely other group, and it's this, for instance, the coelacanths, and it belongs to the sarcopterygians. Sarcopterygians is a sister group of actinopterygians, both from the osteichthyans. And in fact, it is a group that encompasses tetrapods and our own species. So, in fact, the coelacanth is closer to us than it is to a zebrafish, for instance. So, under the name of fishes, we have very different species belonging to very different phylogenetical groups. So it's what is called to be polyphyletic. They don't belong to a single group. And in fact, the name fish that we use a lot, even in science, it's not phylogenetically relevant. And it has many consequences when we interpret and compare data. So it leads to the necessity of investigating a diversity of so-called fish models. And this concerns even teleos species. Here it's point phylogeny of the teleos, which is this type of group. But you have so many species and such a high diversity. It is the largest group of vertebrates with more than 25,000 species. And again, we cannot infer from one species what would be the physiological functions in others. So what is interesting for this diversity of phylogenetical groups represented by fishes is that they give us access to basal features of physiological functions, like access to what could have been ancestral feature of vertebrates with the cyclostome, ancestral feature of the gnatostome, or of the sarcopterygian. Also, it gives us access to evolutionary patterns that are related to radiation of species with this huge diversification of teleos. And finally, they give us access to investigation of impacts of whole genome duplications and gene conservation or gene losses in the evolution of molecular families of interest for endocrinologists, for instance. Indeed, in the evolution of vertebrates, there have been major events of whole genome duplication that likely favored the success of this group. Two genome duplication at the basis of vertebrates, then an additional one at the basis of teleos, and an additional one in some groups, such as, for instance, salmon. This one is quite important when we compare teleos models with other vertebrate models like mammalian models. And in my group, we work a lot on this molecular evolution of endocrine families, hormone, neuropeptides, and receptors. And this is just to give you an example of the history of leptin and its receptor. In human, there is one leptin gene, one receptor for the leptin, one gene for the leptin receptor, so 1 and 1. In the same situation in all tetrapods, and also the same situation in the coelacanth. Also the same situation in the basal actinopterygian, the GAR. But in teleos, following the whole genome duplication, there was an ancestral situation with 2 leptin genes and 2 leptin receptor genes. And that's what we still witness in the basal teleos the yield with possess two leptin genes and two genes for the leptin receptors. But soon after the emergence of these basal groups in the ancestor of glupeocephals, there has been a loss of one receptor. So for instance, zebrafish or medaka, they have two leptin genes, but one receptor. And then later, in the radiation of teleos, there was another gene loss in the ancestor of percomorph, so that some species, such as fugu or sickleback, have only one leptin genes, one leptin receptor, but they are not exactly the same as those in mammals because they are resulting from this duplication and then gene losses. And conversely, in salmonids, following the salmonid-specific multi-genome duplication, there are more receptors and more leptin genes. So that has to be kept in mind when comparisons are made between species. And nowadays we have more and more fish species being investigated around the world for many objectives. It goes from basic biology, aquaculture, aquariology, fisheries, medicine. For instance, zebrafish becoming a vertebrate model for many research in medical sciences, ecotoxicology, evolutionary biology, and more conservation, but also social, cultural values. All around the world, our societies have been evolving together with the sea and the water in the ocean and its resources. So among all these species investigated, I will talk about some laboratory fish models and give a tribute to Professor Richard Peter, who has inspired many of them, including my friend John Chang, with his research on the goldfish. So Richard Peter investigated the neuroendocrine control of reproduction in the goldfish. At that time, it was known in mammals that reproduction was controlled by pituitary hormone, FSH and LH, itself controlled by neuropeptide GnRH. And while investigating where GnRH neurons could be located, he was performing stereotaxic brain lesions. And some of these lesions, surprisingly, led, instead of blocking the reproduction, if it had been damaging the GnRH center, were leading to induce LH release and ovulation. And that's how he first provided the evidence of the presence of an inhibitory factor for reproduction in the brain of the goldfish. Then, with experiments using dopamine agonists or dopamine antagonists, he was able to demonstrate that dopamine was this release inhibitory factor in the goldfish. And further investigation in neuroanatomy and together with lesion experiments demonstrated that these dopaminergic neurons involved in the inhibition of reproduction in the goldfish were located in the preoptic area and projecting directly to the pituitary. So this led to this demonstration of a dopaminergic inhibition of reproduction in the goldfish, in the adult goldfish, beside the classical control by GnRH. This is a double control of reproduction. And this led to many important applications in aquaculture. And since we are in this Chinese endocrinology meeting, I want to emphasize that it went to the application of the what is called the Lin-Pei method from the Professor Lin Haoren from China, so the Lin, and Richard Peter. And the purpose was to use the combined treatment with GnRH agonist and dopamine antagonist to induce final oocyte maturation and ovulation in many fish in aquaculture farms, such as in carps, catfish, or tilapia. But according to the diversity of the teleost, This dual control of reproduction doesn't occur in all species. For instance, it was shown that it's not the case in the cooker, it's not the case in the sibling. So it's not the case in all species where only GNRH is involved. So we followed that study and investigated this question in fish model with some extraordinary life cycle, which is the eel, which was the classical model in the lab where I was at that time. So the European eel, like eels in other parts of the world, like the Japanese eel, have an extraordinary cycle with a reproduction in the ocean. For the European eel, is in the Western Atlantic. A long migration of the larvae up to the continent with the metamorphosis into glass eels before reaching the continent. A juvenile growth, a long juvenile growth in the continent. The second metamorphosis into silver eel and a return to the reproduction site. But when the silver eels leave the continent to perform the oceanic migration and reach the reproduction site, it is blocked at the prepubertal phase. And if the migration is prevented, it will never mature. And this was a question raised many years ago by the philosopher and scientist Aristoteles, who first raised the question of what happens to the eels where we never can find any eggs or sperm. So he was the first one before Christ to raise that question. And it is only in the 20th century that Professor Maurice Fontaine in Paris at the Museum of Natural History was able to induce the development of eel gonads by performing treatments stimulating the gonads, gonadotomy treatments. So he used in the male urine extract of pregnant women, which were known later to contain human chorionic gonadotropin, and was able to induce full spermatogenesis in 1936. And then later, that was not working in the female. And in the female, later, he was able to successfully induce full oogenesis by treating them for several months with carpituary extract. So that leads that the female with the huge development of the belly due to the growth of the ovaries and the male smaller. And this led to the first observation in the world of eel sexual maturation and spanning. This led also to the conclusion that the lack of sexual development in the eel was due to a lack of stimulation by pituitary gonadotropins, which is the situation we see in human species during infancy. So as we were interested to investigate what was the reason of this lack of pituitary gonadotropins. So in human during the infancy, there is a lack of stimulation by GnRH that leads to this blockade of sexual maturation. And in particular, we were asking the question if dopamine could be involved at this stage. In goldfish, it was investigated in adults just before final oocyte maturation and ovulation. So we asked the question if it could be involved earlier at the puberty. So in cooperation with Richard Peter and Unizoa in USA and Richard Peter in Canada, we treated eels with GnRH agonists and dopamine antagonists, and this double treatment was able to stimulate LH synthesis, LH release, vitellogenin production, and increase in the gonadosomatic index. And by neuroanatomy, among all the red dots are dopaminergic neurons in the brain of the eel, and among them we find dopaminergic neurons in the prooptic area that were projecting to the pituitary. Here are the dopaminergic projections, and providing the anatomical basis for this inhibition. So in the eel, we could demonstrate that before puberty, there is a blockade of this sexual maturation due to a lack of GnRH, exactly like in human, but in addition, a strong inhibition by dopamine. And this, as long as the migration to the ocean is not performed. And again, the diversity of situation in teleos shows that in some species, was shown later by a group in Australia, in the mullet, there is the same situation, the puberty. Dopamine is involved in the blockade of puberty, but it is not the case, for instance, in the sea bass or in the sea bream, where only generation is involved in this situation. And so we but also for seaweed, which was raised in lagoons close to the coast, and recent investigation show that it would date back at least 3,500 years before Christ. So we have a very long history of agriculture around the world. And since very early agriculture of carp or tilapia, in many countries, there has not been so much diversification for many years. So as it's shown in this table, the common carp is raised in 48 countries, a tilapia 45, rainbow trout 45, and it's around the world, and which leads to problems such as introduction of non-native species that can affect biodiversity and lack of resilience to global change. So everyone is now concerned by the need for diversification of aquaculture fish species. And this diversification is performed with various objectives. One is farming of local native fish species. This is dealing with food security, avoid fisheries, so overexploitation of wild species, avoid introduction of non-native species, and culture fish well adapted to their environment, and also in the frame of environmental safety. So all these objectives is towards a sustainable aquaculture. And we are, for instance, working in cooperation with groups in South America and Africa to develop the aquaculture of these big fishes that are representative of the basal teleos groups, osteoglossoma. They can become very huge fishes, and that are nowadays very good candidates for aquaculture in South America and Africa. So another objective for diversification of aquaculture is to culture fish species for aquariums. There are many big aquariums around the world showing fish species to the public. There is also a lot of private aquariums at home, but many of these species are not reproduced. So many of these species are still caught in the wild, and there is a very important need for sustainable aquariology. So there are a lot of research for performing closed cycles of fish presented in aquariums. And last but not least, one main objective of diversification of aquaculture is the conservation of endangered species. Indeed, fish species are submitted to the impact of global changes, including not only overfishing, but habitat destruction, obstacles to migration for the migratory fishes, pollutants, global warming, water acidification, changes in ocean currents. And going back to the eel, there has been a dramatic decline of the population in the past decades. So now, for instance, for the three species, temperate water, European eel, Japanese eel, American eel, their stock is a few percentage of what it was in the 1950s. They are classified as critically endangered. And in Japan, there has been a tremendous effort to close the life cycle of the eel with success. And based on experimental maturation, following the protocols developed by Maurice Fontaine in France. And for the Japanese eel, now the cycle has been closed, but there is still a lot of improvement needed due to variable quality of eggs, low success rate, and still far for large production. For the European eel, the situation is worse. The life cycle has not yet been closed. For the moment, it's just this larval stage that has been reached, and there is a huge need for more research. And just in September, we were happy to launch a cost action. It is a cooperative science and technology action, joining all European research laboratory volunteering for working on eel to try to advance this question. And to give you some other examples of the emergency need for conserving endangered species in Europe, in France, namely, says the sad story of the European sturgeon. The European sturgeon is a beautiful large species of sturgeon that was living in all big rivers of Europe and recently was found only in this Garonne river, so in the southwest of France, after its disappearance from all the other rivers. And today is considered like quasi-extinct. It's, as always, a terrible story of taking regulations too late. The decline was observed already largely in the 1960s, and it's only in 1982 that the species was put on production, which was likely very late. And today, there are investigations to try to conserve the species, close its cycle for restorative aquaculture, ex situ conservation, and restocking. Another example is the paradox of the salmon. The production of salmon, Atlantic salmon and salmon species, by aquaculture farm is rising tremendously. But in the same time, the wild population are dramatically declining. And for instance, in France, again, the map of France, that was the river where we could find salmon in the 1,700 years. So we could find salmon all over France in small river like in Brittany and in large river. In the mid-1800s, already there was an edge river with salmon, and today there are salmon in small rivers, and only this is the Loire Angers River, that's the only one left among the long river that has salmon. And in fact, the population of long river are very different of those of the small river because they are able to perform this huge up and then down migration. They don't have the same properties. And again, it is a story of reacting too late. For instance, here at the dam of Vichy, 100,000 adults were counting in the late 19th century to come back every year to reproduce. And today it's only 100. And the ban of fisheries was again established very late. But there is a conservation plan and a farm devoted to conservation of this last and endangered population of long river Atlantic salmon. So I will be short. So now we have cutting edge technologies for promoting new aquaculture fish models. And again, with some historical view, it was a German scientist, Stephen Jacob, that first promoted artificial fertilization in trout farm. Now we have preservation of fish pearl by cryopreservation, but more importantly, preservation of gonies, and based on the pioneer work of the Japanese book of Goro Shizaki and his co-workers, this ability to use gonial cells from an endangered species, introduce them into other species, and producing fish using this other species. So this is a very important cutting-edge technology to preserve endangered species. But cutting-edge technologies for ex situ conservation are okay, but for in situ conservation and restoration of endangered species, we need restoration of habitats and ecosystems. And we are here, human, it's the Roman time mosaic with all the fish species, and we have to consider this concept of One Health, which means that human health is completely linked to animal health and ecosystem health. Thank you. Thank you. For eels, migration triggers the production. So it's a very good question, and there are research conducted on this. So ourselves, we have been working on the effect of high pressure because it is shown that the eel dive deep. There are works performed in Holland on the fact of swimming. So long-term swimming also produces some stimulatory effect. Each time it is a small effect. Seawater is improving. There are current studies on the effect of magnetic field because it is investigated now that they follow a certain magnetic field to reach the spawning ground. But isolated all these factors are not enough, but it is a way to do also the research to try to find what it should be. It's probably all these factors during the migration and at the spawning ground. It's of course for the final save the pheromones. But it's for sure one of the research to find sustainable solutions to the problem.
I have a question. And you also mentioned the tedious as a third and fourth original duplication.
The tedious at the basis of the group, there is a third original duplication. But interestingly, there are studies on fossils of tedious, and they look at the size of the cells in the bones. And in tedious, the size of the cells in the bone is kind of proportional with the size of the genome. And so we can trace back the moment of the third whole genome duplication even in fossils of tedious. So anyway, tedious are special for this whole genome. And so, but like salmonid or some carp have an additional one, but much more risk.
Thank you.
Very great talk, Sophie. Just wondering, have you looked at the peptidergic GNIH system in the eel?
Yeah, but amazingly, yes, we have looked at it, and it's inhibiting in some species, not always inhibiting, but in the eel it's inhibiting. So we were hoping for some factor that stimulate, but it's like this classical discovery by
Okay, it's the end of this session. Thank you so much. Great. Whatever. And then further. Go to now.
With 2 minutes after time, so get started. So my name is Yunbo Xi. I'm a co-chair of this session with Dr. Zibin Zhang, and we decided to divide the duty. So I'll introduce the first 2 speakers, and third down will introduce the last 2 speakers in this session. So we should get started. And the first speaker is Dr. Pei-San Chai from University of Colorado Boulder, and she will talk on functional characterization of a levothyroxine proteasome GnRH related signaling system. Thank you.
Thank you all. Good morning. First, I'd like to start off by thanking Professor Zhang Qingfeng and Professor Zhu Yong for, as well as the local organizing committee, putting together this very wonderful event. I am a Taiwan-born native, so it's always a pleasure for me to come home, and it's extra special for me to be able to speak here. For Gaoxiong, yes. 大概在 So today I would like to talk about a project that has begun about 20 years ago. And this project is not going anywhere. Right? All right. So this project involves the study of a very small peptide that is doing a big job. So this peptide is called GnRH, gonadotropin releasing hormone, and this particular peptide, I think, needs a whole lot of introduction. Is because GnRH is absolutely critical for vertebrate reproduction. Without GnRH, gonadotropins cannot be secreted, and gonadomaturation cannot occur. I'm sorry, I seem to have difficulty handling the pointer, so please bear with me. So without GnRH, gonadotropins cannot be secreted, and gonadal maturation cannot occur, so the animal goes sterile. the importance of GnRH in vertebrate reproduction cannot be overstated. So, regardless of how important GnRH is for vertebrate reproduction, at the beginning of the century, specifically in 2001, our understanding of how GnRH has become a reproductive activator critical for vertebrate reproduction was very limited. So in 2001, that was the state of our understanding. Right here is a very simple phylogenetic tree. It shows that the bilaterians are subdivided into protostomes and heterostomes, and protostomes are further subdivided into lophotrochozoans, such as the flatworms and the mollusca, and ecdysozoas are further subdivided into these molting animals. And at that time, all we knew was that GnRH, vertebrate forms of GnRH, or chordate forms of GnRH, were only present in phylum Chordata. And at that time, some people suspected that GnRH may be present in the protostomes, but we had no solid evidence to support that hypothesis at that time. So fast forward to a year later, that was in 2002, just a year later, Minakata's group in Japan had come up with a very seminal discovery. That is, in the lophotrochozoan, in the mollusk, they found a GnRH-like molecule in the octopus, and they call this GnRH-like molecule octopus GnRH. So when you line up octopus GnRH with a prototype vertebrate GnRH, which is mammalian GnRH right here, you can see a lot of structural similarities between these two molecules. Of course, we have the signature pyroglutamyl residue in the N-terminal and the signature proline amidated glycine at the C-terminus. And in addition, there are several key amino acids that are conserved between the octopus GnRH and vertebrate GnRH. But there's one very notable structural difference between these two GnRH forms, and that is mammalian GnRH, as well as all other chordate GnRHs. They're all 10 amino acids long. They're all decapeptides. However, octopus GnRH decides that it wants to be 12 amino acids long. So this is a dodecapeptide. So, okay. So after that particular discovery, we got very excited. However, we were also a little bit concerned because octopus GnRH is a very short peptide. And at that time, it was only discovered in octopus. So we thought, well, perhaps there's a possibility that octopus GnRH arose through convergent evolution. And what if it's only found in the octopus and we cannot find it anywhere else? So to allay our concern, what we decided to do was to find a second GnRH in the mollusk. So our hypothesis at that time was that if we could identify another GnRH in the mollusk, that would firmly establish the presence of GnRH in the lophotrochozoa. Therefore, it would show that protostomes definitely have GnRH. And if protostomes and deuterostomes both have GnRH, that would suggest that GnRH arose very early on, about 670 million years ago. And by the virtue of it being so important for reproduction, it persisted in all these different lineages, okay, without any functional change, because it's so important for reproduction. It just cannot change its function. Of course, we were completely wrong about that. But back then, we didn't know how misguided we were. And sort of motivated by this misguided hypothesis, we decided that we want to look for another GnRH-like molecule in the mollusk. Okay, so the molluscan model that we chose was a gastropod. This is Aplysia californica. Okay, this is a very well-known neuroscience model because it has a central nervous system and a bunch of neurons that are really well described functionally. And we had always assumed that GnRH would be a neuropeptide. So a better model to study a neuropeptide than a neuroscience model. I just want to point out that a lot of times when I present this photo, students will ask, why are you feeding Aplysia ramens? So these are not ramens. This Aplysia is laying eggs. So these are the egg strands that come out of the side of their mouth. So we decided to search for a GnRH-like molecule in Aplysia californica, and after a while, we did find it. This particular GnRH-like molecule, we call that Aplysia GnRH, or AC, or Aplysia californica GnRH. So when you line it up with the original octopus GnRH, you can see that these two peptides are totally related. The N-terminal amino acid, 9 amino acids are completely identical. The difference is in the C terminus. And what we found that was surprising is that Aplysia GnRH is not 10 amino acids like the vertebrate GnRH. It's also not 12 amino acids like the octopus GnRH. It seems to have a mind of its own. It's 11 amino acids. It's a dodecapeptide. It's a decapeptide. So at that time, we decided to do a lot of functional studies on this Aplysia GnRH. We studied physiologically what it does to the animals. But after several years, we found another GnRH-like molecule in Aplysia californica. And we named this particular peptide Aplysia AKH. AKH stands for adipokinetic hormone, and I'll explain that in just a little bit. So this Aplysia AKH, when you line it up with Aplysia GnRH, you'll find that, again, a lot of the signature amino acids are conserved. And in fact, this tryptophan residue right here is completely invariant. And you'll find that in the middle, there's also several invariant amino acids that are conserved in Aplysia AKH. Well, Aplysia AKH is not a novel molecule at all. It turns out that we named it AKH because it is very similar to another group of peptides that have been studied for decades in the arthropods. And this group of peptides is called adipokinetic hormone. That's why we name Aplysia AKH adipokinetic hormone. So shown right here is a sequence of Drosophila fruit fly AKH. Again, you can see all the familiar amino acids that are conserved. And so Aplysia AKH, we ran it through a lot of checks, and it does satisfy a long list of criteria in order to name a peptide AKH. It has basically satisfied all the criteria. So at this point, our heads were spinning, and we're like, well, Aplysia GnRH actually looks like vertebrate GnRH, but it also looks like AKH. And an AKH definitely looks like an AKH, but it also has some similarity with GnRH. So who is who? They just don't look exactly like one or the other. They're just kind of similar to one another. So what we decided to do was that we formulated some of these sort of principles. We thought that vertebrate GnRH, molluscan GnRH, molluscan AKH, and arthropod AKH seem to all be related. And the operative word right here is because we all know how difficult it is to perform phylogenetic analysis on short peptides. These are the peptides written 8 to 12 amino acids, which just do not have sufficient statistical power to infer their evolutionary relationship. So what do we do to understand how they evolve and how they are related to each other? That requires understanding of their receptors. We have to analyze the receptors and how the receptors are related, because receptors and ligands always co-evolve with each other. So we started to look for receptors for both Aplysia GnRH and Aplysia AKH. And to make a long story short, we did find them both. This is to show you a quick deorphanization scheme of the deorphanization data of the receptor for Aplysia GnRH. We call this Aplysia GnRH receptor. This is just to show that Aplysia GnRH receptor comes in 2 different versions, the long version and the short version. The short version has a N-terminal truncation of 76 amino acids located in the extracellular binding region. So not very surprisingly, the short version is completely inert. If you try to compete up its binding with increasing concentration of Aplysia GnRH, there's basically no competition. But the long version is a full-fledged completely functional GnRH receptor, it responds to increasing concentrations of aplasia GnRH by displacement. And this is just to show you that this aplasia GnRH receptor is completely specific to aplasia GnRH. It cannot be competed out by increasing concentrations of aplasia AKH. So this is a aplasia GnRH receptor that binds very specifically to aplasia GnRH. It does not interact with aplasia AKH. Okay, this is just to show you that the converse is true for aplasia AKH receptor. We have found an aplasia AKH receptor that will be displaced in terms of binding by aplasia AKH, but its displacement by aplasia GnRH is very minimal, even at the highest concentration. So this seems to be a very specific system with aplasia GnRH receptor binding to aplasia GnRH and aplasia AKH receptor binding to aplasia AKH. And just in case if anyone is interested, both receptors, when they're activated, they increase the accumulation of inositol phosphate. So basically, both receptors are GQ coupled. Okay, so now we have found the receptors for both aplasia GnRH and aplasia AKH. We can start to perform phylogenetic analysis. In cooperation with Stacia Sauer and David Placheski at University of New Hampshire, we perform phylogenomic analysis in order to try to infer the relationship between AKH receptor, GnRH receptor, and vertebrate GnRH receptor. So note that this is phylogenomic analysis, not phylogenetic analysis. This basically takes all the genomes, available genomes in the database, and perform the analysis instead of picking and choosing specific sequences to perform the phylogenetic analysis. So right here, I want to point out that there are three groups of receptors that are interrelated. We have GnRH, caudate GnRH receptor, we have AKH receptor, and we have another group of receptor called the corazonin receptor, which I'll talk about in just a little bit. But aplasia AKH receptor binds to an AKH, and it also clusters with other arthropod AKH receptor. So I think it's safe to say that aplasia AKH probably is a real AKH. The problem that we experienced was aplasia GnRH. Aplasia GnRH binds to a receptor that does not cluster with AKH receptor, also does not cluster with the true GnRH receptors, but it clusters with a group of receptors that are called corazonin, okay, or CRZ receptors. So it seems like we have another member of the GnRH-like family that it has forced its way into the GnRH-like peptides. So let's talk about exactly what corazonins are. So shown right here are some of the sequences that I have already presented to you. We have octopus and aplasia GnRH, aplasia AKH, and fruit fly AKH. So on top of that, I'm going to add corazonin. So corazonin, like AKH, is not a new peptide. It's not a new hormone. Its presence has been studied, again, for many decades in arthropods. Shown right here is a sequence of honeybee corazonin. And again, you see the familiar motifs: pyroglutamyl residue, invariant tryptophan, and the tyrosine right here and the serine right here, very well conserved in some groups of the GnRH-like peptides. So you look at the molluscan GnRH, you're like, what are they? Are they true GnRH? Are they corazonin? Are they AKH? Well, I think we basically answered our own questions. When we did the phylogenomic analysis comparing the receptor sequences, I think we can pretty comfortably deduce that aplasia AKH is a true AKH because it looks like an AKH. It binds to it receptors that cluster with arthropod AKH. So it should be appropriately named AKH and it should not change. But the problem comes with aplasia GnRH because aplasia GnRH binds to receptors for corazonin. And I think because of its ambiguous similarity with vertebrate GnRH and AKH, it causes a lot of confusion. But based on receptor analysis, I think Aplysia GnRH should really be renamed as Aplysia corazonin. And that goes also for the octopus GnRH, as well as a lot of gastropod GnRH that have recently arisen. So at this point, one might ask, well, if corazonin and AKH have been so well studied in arthropods, exactly what do they do? Well, just shown here are some examples of what corazonin and AKH can do in arthropods. They are truly multifunctional. They can do basically a little bit of everything. So shown right here, I have some examples of what corazonin does in arthropods. They are cardioexcitatory, and basically they were originally named because they stimulate the heart, corazon, okay, in the cockroach. That's why they're originally named corazonin. They can also regulate metabolic functions by regulating insulin production. They can perform reproductive functions by coordinating sperm transfer. They can initiate ecdysis, and they can also stimulate pigmentation. Okay. When you look at AKH, the functions of AKH in arthropods are just as diverse. They can mobilize energy resources like carbohydrates and lipid reserves. Basically, AKH is a catabolic hormone that breaks down energy reserves in order to supply energy for movement. AKH can modulate immune and stress response, and AKH can also stimulate pigmentation in arthropods. Very diverse array of functions. So what does AKH and corazonin, what do they do in Aplysia? So when we looked into the function of Aplysia corazonin and Aplysia AKH, we found something quite different from what they do in arthropods. In Aplysia, corazonin stimulates motor functions. Immediately after you inject the animals with corazonin, these animals will attach to the substrate. Come to the substrate and fly them out. In addition, they immediately open up their back. There are two little fleshy coverings in the back called the peripodia. They will immediately open up peripodia. So shown right here is an Aplysia that has been injected with a vehicle, just going about its merry way. But once the Aplysia has been injected with corazonin, you see that the peripodia completely open up to expose the gills. It could be promoting respiratory pumping or increased oxygen consumption. Aplysia AKH, on the other hand, does something a little different. Its most important effect is to promote acute weight loss. Okay, and this acute weight loss is done by the loss of blood volume. In fact, 6 hours after the injection of Aplysia AKH, these animals lost about 30% of their body mass. And that loss is through the extrusion of their hemolymph, of their blood. It's a tremendous loss of blood volume that is really kind of atypical for any peptide to exert. And right here, this is to show that we have a running joke in the lab that if you want to be beautiful and lose weight within a few hours, just inject yourself with some Aplysia AKH. The other thing that both peptides do is that they both inhibit feeding, with Aplysia AKH performing most of the feeding inhibition, and is much more powerful as an anorexic peptide than Aplysia corazonin. And of course, the question everybody asks is that, do these two peptides stimulate reproduction, like Gerber-G and RH? We have looked. We would love to find some reproductive association between these two peptides and reproduction, but we have looked at all these different effects: egg-laying behavior, egg-laying hormone secretion, gonadal development, immunogenesis, gene inversion. The answer is a simple no. We cannot find any reproductive stimulation exhibited by these two peptides. So we formulated our hypothesis. This is currently what we think. We think that a GnRH-like ancestral molecule emerged 670 years ago, 670 million years ago. And this ancestral peptide, through the course of evolution, has diversified in both function and structure tremendously and performed multifunctional roles as cortisone, AKH, another related peptide that I did not talk about called ACP. So these GnRH-related peptides, they're no longer associated with reproduction. They can be associated with reproduction. Don't get me wrong. They can absolutely be reproductive, but they don't have to. Only in phylum Chordata, in animals possessing a real pituitary, does GnRH become recruited to stimulate gonadotropin secretion, therefore assuming the role of central reproduction. So here are the conclusions. The GnRH superfamily includes all these different diverse peptides. They perform very diverse functions that are taxon specific, and Aplysia has both AKH and cortisone signaling. And thank you so much. And these are the people who have contributed to my research. But I also want to make a plug for University of Colorado Boulder. Although I could perform research on seawater creatures, where I reside, it's completely landlocked. But we do have nice mountains. So these are the Rocky Mountains, and the university is nestled at the foothills of Rocky Mountains. And my lab is located right there. So I welcome everybody to visit, see, and discuss comparative endocrinology. Thank you.
For a few questions.
Yeah, very nice talk.
Thank
So,
you.
two related questions. So, what controls the regulation the AKH GnRH, like insulin? The second question is, have you looked at whether those AKH GnRH control the ovulation and maturation?
Right. So thank you. What controls AKH and cortisone? I will say first about AKH. AKH is a volume regulator, so it exudes body volume, body fluid after administration. We found that body volume is a very powerful regulator of AKH level. When you swell the slug, the Aplysia up by a hyposaline challenge, all swelled up, AKH expression actually goes down. So what we think is that continuous levels of AKH is required to keep the water flowing outward from these animals, because these animals are basically like a bag of water, so you kind of keep getting the water out. So AKH is very sensitive to the water amount in the body. Cortisone, we don't quite know yet. And sorry, the second question is maturation. We did look at maturation of the ovotestis. There's absolutely no effect. Yes.
Yeah. So for this sugar family, GI sugar family, they induce vertebrate
So the original study isolated octopus GnRH. What they did was they used octopus GnRH in an in vitro pituitary assay, and they found that octopus GnRH at very high concentrations could, in fact, stimulate LH secretion. And some people, have used that pituitary gonadotropin secretion assay to test whether or not these invertebrate GnRH-like molecules could bind to vertebrate GnRH receptors. And it's true that in some cases that could happen. So there is definitely some conformational similarities between them. Yes.
So I have a question here. So you just mentioned AKH is small. On its own, it actually causes a loss. But do you see that what hormone they will generate when you actually simulate
Yeah. So that's a good question. Initially, we thought that injection of AKH would just move the water out. Okay, everything remained. Okay. So later on, we found that that's not true. Injection of AKH actually gets rid of everything in the blood. The ions, the proteins, the hemocyanin, that's what they had, not hemoglobin. hemocyanin. So there is something really strange going on. It's not packaged with water, not just the water. And it lasts, this decrease in blood volume lasts for about 48 hours after the injection. After 48 hours, they will gradually repeat their blood.
Some time. So our next speaker is Dr. John Chen from University of Alabama. He's going to present PI3K and ERK-dependent signaling differentially contribute to acute basal and agonist-limited LH secretion and long-term basal LH secretion and availability in goldfish pituitary cells.
Well, thank you very much for inviting me to present this talk. It's hard to hold two great talks, so it's hard my best. So my lab has been trying to look at the neuroendocrine regulation of pituitary hormone secretion using goldfish as pituitary cells as a model. And today I'm going to talk a little bit about the regulation of LH secretion from the LH cells. And in goldfish, the control of LH secretion is multifactorial with two fish GnRHs. One called initially classified as salmon GnRH because it was discovered in salmon. The other one is chicken GnRH2 because one of the two chicken forms, and now it's called GnRH3 and GnRH2. Also, there are also other inhibitory factors, including dopamine that Sylvie has just introduced to you. And then there's also direct innovation by other, including other neurotransmitters and other hormones as well. So my lab is actually more interested in how the activation of GnRH receptors like ghrelin receptors or PACAP receptors would then use different signal transduction mechanisms to regulate LH and GH cell function. Next. Right, thank you. So for most of the experiments, we use dispersed pituitary cells, which get rid of all the neuroterminals in the pituitary. And then once we get the dispersed cells, we can look at hormone secretion through a perfusion system where you can pump media through cells from beads, and then we can collect media for hormone measurements, or we can plate them for long-term studies, and we can also use the dispersed cells for various different kinds of manipulations, such as messenger RNA measurements, Western blocks, electrophysiology, concentration, etc. I'm not going to present data for all these. I'm just going to mainly present hormone secretion data as well as Western blot data today. Now, this is just a summary of some of the signal transduction pathways that we have looked at, including increasing intracellular calcium, protein kinase C, PI3K, MAP kinase, etc. The take-home message is that yes, although intracellular calcium changes are the components for multiple signal transduction cascades, there are also differences. For example, in terms of the utilization of protein kinase C, PKC, in the sense that the GnRH as well as ghrelin uses it, PACAP doesn't, and then the other also highlights differences. And also, more interestingly, even the two endogenous goldfish GnRHs also have differences in terms, for example, of the use of uraconic acid phospholipase A2. So recently, we've been looking at the involvement of PI3K as well as MET kinase in GnRH action. And since PKC is also utilized by GnRH as well as Ghrelin, so we were interested in whether Ghrelin could also involve things like PI3K. So this is just summarizing what I just talked about, the differential use of different signal transduction mechanisms, probably as part of the multifactorial regulation of IL secretion. And then the Ghrelin and GnRHs both uses PLC, phospholipase protein kinase C calcium system. And then additionally, we also have the MECR kinase system in the GnRH, as well as the PI3Ks. Now, the use of phospholipase C as well as the MECR pathway in a typical G protein coupled receptor system, such as GnRH receptor, is sort of a little bit, not expected, because classically, if you look in the right, the right-hand side, the GnRH receptor is classically coupled through G protein, GQ alpha, and then you can activate the protein kinase C phospholipase C pathway. And the MECR mechanism traditionally is associated with receptor tyrosine kinase signal transduction. And then, however, recently, with the discovery of the phospholipase PI3K involvement, again using G protein into the GnRH system, both the G alpha and G beta gamma involvement, and then that would activate PI3K, leading to production of PI3 or PI345P3 classically, and then through the recruitment of pH domain-containing molecules, so those ones can specifically bind to specific phospholipid moieties, they would recruit an enzyme like PDK1 as well as AKT, and then the recruitment of AKT and PDK1 allow for PDK1 to activate AKT, which then have downstream actions controlling cell function, including secretion. Now, interestingly, in the literature, activation of PDK1 can also lead to activation of protein kinase C, and protein kinase C can also then cross over and activate the MECR signaling through activation of the MET kinase reaction. So this is the classical view of how these two branch of signaling pathway can also be recruited through GPCR receptors. Now, in terms of PI3 kinases, in mammalian PI3 kinases, there are three general classes, class 1, class 2, and class 3, and three classes of different substrate preferences can then generate different kinds of phosphoninositide phospholipids, and the class 1 being the typical one that would generate the PI345P3. And there are also sub class within each class. There are also different members. And interestingly, the two GnRHs that we've been studying, yes, both of them do use the class 1 PI3K, but they also use similar as well as dissimilar isoforms of the class 1 PI3K. So one of the questions that I've been asking is whether Ghrelin also uses PI3K, because when GnRH, it uses the phospholipase C PKC system. So to address this question, we used initially the cell perfusion system, which look at the effects of acute release. And we used 4 different inhibitors of the PI3K, including a pan PI3K inhibitor that inhibits all 3 classes. And then we also use a class 1 specific inhibitor and 2 class 3 specific inhibitors. We haven't been able to find a commercially available class 2 inhibitor, so that's what we have. So this is an example of some of the results of the hormone secretion response to Ghrelin. So if you apply Ghrelin, which is the little arrow in the middle, you get a transient increase in IL secretion. And then in columns that we just have application of the PI3K inhibitor LY compound, you can see that there is a slight elevation in basal secretion. And then when you, in the middle of the application inhibitor, you apply your ghrelin, you essentially have a blunted response. So when we quantify the net response, you can see that the response to ghrelin is essentially abolished in the presence of the PI3K inhibitor. And then there is a statistically significant elevation in basal secretion due to the presence of the inhibitor alone. And to summarize the comparisons with the other PI3K inhibitors, I have this table. So the ability to block the LX secretion response to ghrelin is also replicated with class 1 PI3K inhibitor, but a class 3 PI3K inhibitor doesn't do that. However, in terms of basal secretion, the elevation of basal secretion in the presence of inhibitor, class 3, but not class 1, mimic the pan-PI3K inhibitor. So what we can say is that ghrelin-induced LX secretion involves PI3Ks, and class 1 PI3K, but not class 3, mediates the acute effects of ghrelin on LX secretion. We haven't studied it directly class 2. Now, PI3K can also function in the differential regulation of basal secretion versus stimulated secretion. In this case, class 3, but not class 1, PI3K exerts a negative modulatory effect. So if you apply the inhibitor, basal secretion increase. So that's what we interpret as the normal function of this kinase system is to actually exert negative modulation on basal secretion. And likewise, although I haven't shown it, some of the other results that we use in the PIT1, which blocks the interaction with the pH domain containing binding, as well as AKT inhibitor, that also replicates the effects on basal secretion with the PI3K pan-PI3K inhibitor. But again, we don't know the class 2 effects of this one. But the big take-home message from this part is that similar transduction pathways, such as PI3K, differentially participates in the agonist-induced and stimulated LX release, and also can differentially affect basal release as well. So what about the MAP kinase system, MAC-ERK kinase system? If we apply the inhibitor for MAC-ERK, instead of seeing an elevation in basal secretion, we see a suppression. So although both MAC kinase and PI3 kinase are mediating the short-term stimulatory effects of things like GnRH, the effects in basal secretion are different. So not all of these pathways have opposite effects on stimulated versus basal. So they're all selective. So we're then asking, what about longer term functions of these gonadotropin cells? What are some of these effects of the MAP kinases and let's suppose PI3 kinases on long-term basal secretion and long-term cellular LH measurable protein content? So we ask, does PI3 signaling participate and MAP kinase signaling affects those long-term basal release as well as cellular LH content? Now, the phosphorylation state of AKT and ERK can be used as indices of their activity and activation. So we also using Western blot measured what the effects of inhibition of PI3 kinases as well as MAP-ERK kinases on the phosphorylation state of ERK and AKT using Western blot. And these are all now longer-term 2-hour steady incubation studies. So we've seen this diagram before. Interestingly, downstream of PDK1 and AKT stimulation, there's also the recruitment of TOR, targeted rapamycin complex 1, and PDK1 can through mechanisms can stimulate TOR complex 1, AKT can also do that by actually inhibiting two inhibitor-regulated components to stimulate TORAC1. And there's also a TORAC2 complex, which can directly actually stimulate AKT as well as activate protein kinase C. So to look at how some of these signaling components affects the long-term functions, basal functions of the gonadotropin cells, we use various inhibitors, specific inhibitors for different components and different parts of it, as well as also blocking the pH domain interaction using PIT1. Some examples of results. On the upper part of the panel is shown the effects of the pan-PI3K inhibitor versus the specific class 1 PI3K inhibitor. As you can see, the class 1 PI3K inhibitor in long-term hormone secretion, basal secretion, now is elevating instead of having no effect. But however, it doesn't replicate the effects of the pan blockade of all three classes of PI3K on LH cell content as well as total LH availability. And in here, we're showing results of the blocking of specifically the TORAC1 complex versus the combined blockade of both the TOR complex 1 and complex 2. Again, you can see that there are slightly differences, especially, for example, in basal secretion, where you block both of them, has no effect. If you only block one of them, you get elevation. And to just summarize, before that, we're looking at the phosphorylation state of AKT as well as the ERK pathway. Now, you say, okay, well, we're using a lot of inhibitors. So what are they actually doing what they're doing? So if you look at blocking AKT using the AKT inhibitor, you reduce AKT phosphorylation. So it reduces the activation of the pathway. So that works. If you look at blocking MECR using U0126, it reduces phosphorylation, which is fine. However, we notice some unexpected results. If you actually block MECR, you elevate AKT phosphorylation, you activate the pathway. However, also, if you block upstream components of AKT, such as using the PIT1 blocking pH domain or blocking PI3 kinase activation, you also elevated AKT phosphorylation. In terms of the MAP kinase phosphorylation, if you're using the pH domain blocker, it actually reduces. Okay, so there are all these differences. So what's going on? So let's look at all of the accumulated results so far. So notice that acute release in perfusion to a 5-minute pulse of the agonist, and versus, and I mean to acute release on basals, right? Versus long-term steady incubation on basal by inhibitors of different effects. So long-term results are totally different things. If you look at the LH content, it's not always related to whether it has a similar effects on long-term LH secretion, right? So regulation of probably recruitment of protein synthesis and versus secretion are two different things. And these pathways participate in doing different things on these effects. If you look at the phosphorylation results, we definitely have crosstalk between the AKT pathway as well as the MECR pathway. So if you block AKT, it reduces AKT resistance. If you block MECR stimulation, you stimulate AKT. Furthermore, even if you now block the PI3K dependent components, you see different effects on AKT activation. So things are not linear because we have a web. So you've seen this before, this diagram before. In the literature, there's evidence to suggest that activation of nectar activates TORC-C1. And activation of TORC-C1 would exert a feedback inhibitory loop on upstream PI3K activation mechanisms, therefore could eventually then downregulate TORC-C1. And TORC-C1 also feedback onto the macro pathway to inhibit RAS, therefore may inhibit MEP kinase ERK activation, which is stimulatory to TORC-C1. So it's a positive negative feedback loop all combined together here. And then you also have the TORC-C1 can inhibit TORC-C2, TORC-C2 being stimulatory to AKT. So that's another feedback loop mechanism that exists there. If you look at AKT, AKT can also directly cross over to inhibit RAS, which can then inhibit MEP kinase ERK. So with this kind of possible feedback loop, we now have to think about the idea that regulation of hormone secretion and cellular content definitely are associated, and differential use of intracellular signaling components have different processes on these two elements. Acute versus long-term basal secretion are regulated differently. There's crosstalk between the different pathways. And with this crosstalk and possible feedback in intracellular signaling, therefore, you need to think about all of these aspects, especially under longer-term stimulation or longer-term incubation. And because in the goldfish pituitary, there's direct innervation of the pituitary cells by neurons that has neuropeptides or transmitters. Whether those are transient release or long-term release conditions, or long-term, for example, feedback under, well, for estrogen and testosterone long-term feedback, those two are completely different situations. So you have to think about, we should need to think about how the natural system works before we can actually interpret whether the acute release is more applicable to your question or the longer term release. So if you have longer term release, you're going to get into the complexity of feedbacks. And you can, so the typical message is that we still don't know a lot of things, and we need to be careful in how we interpret our data. So the research is done at the University of Alberta, and we have to acknowledge that we're situated on Territory 6, Treaty 6 territories, the traditional homeland of First Nations and the Métis people of Canada. And I would like to thank the organizer. 谢谢.
Thank you, Chen. If I am not wrong, one of the major differences between mammalian LH secretion is the pulsatile secretion in mammals and baby do. So can you get some insight from all what you investigate? Is there any strong differences in the pathways?
Well, the actual use of PI3K pathways and the subsequent pathways in mammalian allergen FSH is really understudied. So there's no real direct comparison at this point, unfortunately. But now there's also some evidence from Professor Lin Hao Ren's lab, as well as collaboration with Peter, that there may be also pulsatile secretion of allergen GH in fish. So the neuronal input is probably also pulsatile in a sense.
So yes, and I just say the complexity of different signatures, as well as the negative feedback. I have another sort of a related question. pharmacological drug. So the concentration, time you use to treat the cells probably would cause undependent on but many of the drugs developed based on these, we do not know, for example, what the cost is. Indeed, only I wonder how would we do that.
Well, part of the way that we're trying to, hopefully we try to address it, is to look at where available proteomics, the molecule that is supposed to be targeting, and the targeting site where they conserved, and where in this instance that we have looked at it, those are all conserved at this point. So that's what we say.
All the function of the species also conserve different members.
But some of these class 1 versus class 3 has inhibitors bind slightly different places, and those places also. So that's all we can say at this point. And then using pharmacology, we always have to be careful too. Yeah. And also one kind of caveat that I need to point out is the phosphorylation assays are done with mixed population cells, not purified cells. That's another caveat.
Any other questions? One has assume that the remaining function for 2 is still.
That's the implication.
Yeah. Did you analyze any downstream based on your diagram or any downstream of 2 to analyze?
Well, the only one that we looked at is AKT phosphorylation. This is one, but not other ones. We can always look at more.
So I'll turn the podium. Yeah. So our next speaker is Dr. Huang Hongkong from the Institute of Cellular and Organistic Biology, Academia Silicon, Taiwan. So the title is Post-Toxin Mediates the Secondary Response of Bile Regulation Epidemic Pitch on the cold stress.
Thank you. My talk, nothing related to the reproduction. First, I would like to thank my co-operator, Dr. Paul, is responsible for most of the experiment, and other postdocs is working on electrophysiology. And Dr. Zhou Mingyi is my co-operator. He will give a talk next. And we know for the thermoregulation, in vertebrates, there are 2 main strategies. One is endothermic and another is hypothermic. Human is endothermic, and under a cold situation, human will increase the metabolic heat production
to keep their body temperature constant, so that can maintain the whole body biochemical pathway function or near the thermal optimum. However, in the case of the ectotherm, they allow the body temperature to shift with their thermal environment. Usually, we accept that the fish exclusively rely on the behavioral strategy for the thermal regulation. However, in many papers, they also found that the fish upregulate some physiological pathway to overcome cold. For example, they will remodel the cellular pathway, for example, through the mitochondrial genesis, ion transport, or some activity to regulate the biochemical flux against the change in body temperature. And okay, here as you can see, here show the very complex network with the thermal regulation in human. You see the POA is the area in the hypothalamus is receives the signal information from the different sensor receptor. So in various organ, and then in this area, sorry. Okay. So the information will integrate and the process related to the downstream thermal effector for the physiological regulation. For example, for the ground tissue thermogenesis and the cutaneous vessel motion, and also some kind of evaporation for cooling. Okay. And then if we look at all this hypothalamus, POA is over here, but if we see the two areas near POA, here is SON and the PVN, they contain isotocin and vasopressin neuron. And we know isotocin and vasopressin is involved, a very important hormone involved in the water and ion homeostasis in human. So I see. Okay. So if we can look at the detail of this neuron circuit, you can see in the PVN and SON, there are some thermal and osmosensor neuron. And the integration between the thermal regulation and body fluid balance maybe exists in the human. So specifically, when in the human some hypothermia appears, then the evaporative cooling is an important way to down the temperature, down the body temperature, and then this will result in the hydration and also some kind of the disturbance to the body fluid homeostasis. So for compensation, vasopressin will be stimulated to regulate water retention for compensation for recovery of the body fluid homeostasis. So in some paper, they also mentioned the release of the vasopressin or oxytocin will increase if we give some temperature stimulus. If we by a simultaneous increase the temperature of the osmolarity, the release of the hormone will further increase. So this demonstrates some kind of integration between the thermal regulation and body with regulation in the human. So how about the fish, the ectothermic actually, some very old paper also mentioned post the POA also show some temperature sensitive neuron similar to human. However, nothing is known until that paper. So there's actually we have been interested to this issue maybe more than 10 years ago. So we conduct some study to look at this issue. So first, I would like to introduce the fish arm regulation. You see, the fish, instead of the kidney, is a major organ for the body with acid-base ion homeostasis. And in the deer, you can see some kind of cell we call the ionocyte. They have a lot of the mitochondria use ATP to drive various transporter for active transport. And we identify different kind of ionocyte here. It's similar to the different kidney tubular cell in different segments. And then they express different type set of the ion transporter responsible for transport of different ion. Like this one is for the chloride uptake. This one is for the calcium uptake, and this one is for acid secretion, sodium uptake. And we also look at the action of the different different hormone on the differentiation, expression of the transporter, also the function of the ionocyte here, you can see. And then among these hormones, we particularly look at the action of the isotoxin. Isotoxin is the fish of the human oxytocin, and we found he has a positive regulator for the ion uptake and acid secretion in fish. Just to show you quickly show you the result the evidence we got. Here you can see when the fish treated with distilled water is a water lake of ion, and this is straight to the fish, and then after acclimation, the isotoxin expression is stimulated. And we also found that the same here. A different kind of environment will affect the expression of the oxytocin receptor. We also look at this receptor localize some sort of the ionocyte fish here. And here we just use a morpholino knockdown for the overexpression way to do the effect of the loss or gain of the function of the oxytocin on the ion regulation function. You can see knockdown of the oxytocin, the concentration, ion concentration was suppressed. Okay, and also we can see the density also of ionocyte will decrease. On the other hand, if you inject the cRNA of the oxytocin, will increase the number of ionocyte. And here show the in situ hybridization signal of the different transporter. You see, not loss of function of the oxytocin will decrease the number of the transporter. And we also see the knockdown or loss of function or gain function will affect the number of the FOXI3A label cell. The FOXI3A is a transcription factor related to differentiation of the ionocyte. And also the P63 is a marker for the stem cell of ionocyte. They also affected by the loss of function or gain of function of oxytocin. So the conclusion is that oxytocin is regulate the differentiation, proliferation, differentiation of the ionocyte, also the expression of the transporter, and eventually affect the function, ion transport function. Actually, we also look at recently. We also look at the resorption effect on the ion regulation. We found the similar result is rather different. So I think I skipped the detailed result. So you can see resorption also affect the differentiation of ionocyte, also affect the expression of related transporter, but only exert their action on the certain type of ionocyte, and also eventually regulate the function of ion transport of acid secretion and chloride uptake. What kind of detail? Anyway, and then we started to look at the physiological mechanism of fish acclimation to a cold acclimation about 10 years ago. And we know the zebra fish can survive very well in the 28 degrees C. We also acclimated they to the low temperature as low as 12 degrees C. They can survive very nice. And then we did the microarray analysis to compare the gene expression profile between the control temperature and the low temperature acclimation for 1 month or two weeks, and look at this is the difference in brand and the gear. And then first we found when the fish transfer to the 20 in the normal temperature, they show the certain amount of the calcium uptake is very important for their calcium homeostasis. If you transfer to 12 degrees C, right away they decrease their calcium influx. But after 30-day acclimation, they can upregulate their calcium influx to the level of the control, so that totally, completely compensation. Okay. And if we look at the detail, we compare the 28 control, the low temperature, you can see this cell, some kind of cell number largely increased. One is the calcium secreting, that is the ECAC, calcium channel is a transporter for the calcium uptake. Another one is the carbonic anhydrate is a marker for the acid secretion cell, also increased. And as I mentioned in the previous slide, plus 3A label cell also increased. This is a precursor of the ionocyte. So that means the compensation involved in the ionocyte differentiation and expression of the regulation. And then we, from the microarray analysis, we found very interesting data. Here we found several genes. This is cold inducible 1940, something like that, and some other, I don't know the detail about this molecule. All these molecules are increased, upregulate after acclimation to low temperature. However, two gene is very very interesting. Some of them, one is the POC, another is the OTP. And then we searched the literature. We found that actually there are a serious study on the zebra fish. Look at the differentiation of the oxytocin neuron, and here it shows the very complex network and the different kinds of the transcription factor involved in this pathway. And then it looks like the OTP is a central of this network. And then we examine gene expression between two groups of the temperature and all the related transcription factor. And we found most of the transporter, this transcription factor will stimulate it after acclimation to low temperature compared to the control group. So that really exciting. And then we did the loss of function of OTP the central transcription factor for the differentiation of the isotoxic neuron, as I mentioned. And then all the related transcription factors expression were downregulated. And more important, the transporter, related transporter in the ionomer site, all downregulated after we did the morpholino knockdown of the OTP. So that raises a possibility. There must be some connection pathway from the OTP and then oxytocin neuron and the ionocyte and the ion transporter and the ion transport function, some kind of the pathway here. And based on this data, we propose oxytocin may mediate the compensatory response of ion regulation in fish for relation to a cold space. And after that, just 2 or 3 years ago, we are lucky to get some budget support. So we have another cooperation with Dr. Zhong Linyi. We started this work. And now, first, we look at those expression of those oxytocin neuron-related transcription factor expression after acclimation to low temperature for 1 week this time. And then you see most of them still you can find is upregulated consistent with the previous 10 years ago study. And then we use oxytocin GFP9 to show the oxytocin neuron in the brain of the fish. We compare the 2 groups. As you can see, the green signal C show the GFP signal of the oxytocin. You can see some oxytocin neuron here in the two group brain, and we also how to say it? We also did the C4's in situ hybridization to indicate the activity of this neuron. And then you can see here the this merge the signal of the C4 and the oxytocin. You can see the number and the activity of the neuron is higher in the low temperature group compared to the control group. Okay. So this suggests low temperature will stimulate the differentiation of the oxytocin neuron, also stimulate the activity of the oxytocin neuron. Okay. And then if we want to connect, to demonstrate this connection, we should look at the oxytocin receptor in the ionocyte. So then we can say they show the direct action on the ionocyte and their function. So here we did the double in situ and the histochemistry. Here show the green signal, see the 2 kinds of the oxytocin receptor. You see the proton pump the marker for one type of ionocyte, NKA is another marker, another ionocyte. You see the double colloquial signal here, both receptors. Thank you. And then again, we see the gene expression of the transporter after acclimation to low temperature for 1 week, and we found only at the 7th day after acclimation, some of transporter expression and the receptor expression were stimulated after acclimation for 7 days. And finally, we did another functional analysis, and we use the oxytocin agonist is a carbetocin. Use this, we treat with the lava because we have a technique, electrophysiological technique to measure the ion transport function in intact embryo. And so we in this experiment, we incubate larva with agarist for maybe for 30 minutes, and then transfer to the low temperature and normal temperature. And here show their effect on the acid secretion in zebrafish embryo. You see this control group and low temperature group will suppress their acid secretion function. If you add the agonist and then they show some kind of recovery, dose-dependent recovery of the acid secretion function, demonstrate the action of oxytocin for the compensatory response in ion transporter in zebrafish embryo. So I think our data also, we still have to do more experiment. I think we'll data so far we collected can support our hypothesis that when the temperature decrease, they will induce will impair the zebrafish body fluid disturbance in homeostasis, and this gives some signal through the receptor, osmotic or chemical receptor, and then in the brain, and then they will give the signal to the OTP, and OTP stimulate the oxytocin differentiation, and then release more oxytocin to the bloodstream and reach approach the gear through the isotopic receptor. They stimulate the ion side differentiation, stimulate the expression of ion transporter, and then eventually increase the ion transport function for the compensation for recovery the your body for ionic homeostasis. So, and I think we still have several issues to be studied in the future. First one, we should study other fish species. You know, there are so much fish, so many fish. And we also tend to consider whether the oxytocin also involved in the energy metabolism in fish under cold stress, because we know in human, oxytocin is related with some kind of thermogenesis in the human study tissue. Another one, the vasopression, is another issue we should study because they show some kind of similar action as oxytocin with other difference. So I think this is the end of my talk. Thank you for your attention.
And we still have few time. We still has yes.
For the OPT portfolio, did you look at the neurons and see that the human cells are actually so you have the new tools now. Yes.
Really nice work. I have two questions. The first is regarding the first project. That's what you see a chain of things. The normal model goes to stem cell. We talk about stem cells. My question: which that? So,
I think the stem cell. I cannot say it. I don't have data, but I think it may be a different because we know the model for the model of the ionocyte differentiation. There are many each other show different actions on the ionocyte from the differentiation. So no, maybe some kind of indication.
Receptor expressed in the space. To show the receptor expressed in the mature audience. Yes, those they also expressed. I also saw one of the slides using P63 staining, and it shows the number of cell changes. But look, the size also changes dramatically. Cell size change.
Surface area, the embryo limited. So if you do number increase, you send it. Otherwise, space for it.
The second question probably related to this question. We show one of the oxytocin GFP neuron. We say that neuron GFP neuron number decreases in phospho activity changes. I agree. It also looks like the synaptic connections between this neuron actually increases. You have a chance to look at that.
I did not notice the detail. Maybe Dr. Zhou can mention it.
You mentioned to talk about oxytocin is acute response for stress.
I don't think so. I think both, maybe both. In the short term,
Long term,
in the short term,
you have 9 hours. Will you do some experiment between 9 hours and 30 days?
you ask too much.
Okay. So, let's say, okay. I think
Among the transporter or ion channel you have been investigating, there is TRPV6, which is the temperature TRPV6 I saw on your slides, which is the temperature sensor in this report. So do you think there is a temperature sensing in your system?
Kind of the isoform.
Yeah, but do you give the name TRPV6, which is the name also in mammals of the temperature sensor, or not only TRPV? So just because in your scheme, you mentioned chemoreception, baroreception, but you never mentioned thermoreception.
So our last speaker of this section is Dr. Zhou Yi. Maybe the collaborate. He's from Department of Cell Life Science, National Taiwan Institute, Taiwan. The title is Consortium Neural System for Ion Regulation in Fish.
Thanks for the kind introduction. So I will also talk about the oxytocin and reciprocity system in zebrafish. So this beautiful picture is indicated in oxytocin and reciprocity neurons in the hypothalamus of zebrafish brain. So according to the previous speaker, Professor Huang, indicated that the code in stimulates the differentiation of oxytocin neuron and releasing of the oxytocin. Then the ionocyte express the oxytocin receptor. Then that process can compensate the ion loss after the code formation. So if you check the oxytocin neuron under the control and code environment, you can see the cell number, oxytocin cell number is increased. But our data also indicated that not only the cold environment. So if we put the fish into the deionized water, that means hypotonic or iron deficient water, the number of the oxytocin neuron also significantly increase, indicating that the oxytocin itself, this neuron is responsible for the iron regulation and acid-basic balance, according to the previous speaker, Professor Wang Hong. So when discussing the oxytocin system, always the other neuropeptide neuron also mentioned that is vasopressin. So we actually inject the morpholino into the zebrafish embryo, and we found knocking down of the vasopressin also impair the iron and the acid balance. So the chloride content and the calcium content, chloride content was decreased, and the acid secretion function also impaired by the morpholino. So in the embryo injected with of the vasopressin neuron, morpholino, the two types of the ionocyte, like called NCC, sodium chloride transporter, and proton pump rich cell also decreased. So that means that the vasopressin itself probably acts as similar actions as the oxytocin neuron. So it led our lab to study these two neural circuits, oxytocin and vasopressin. So to do so, we try to generate a transgenic fish, in which the oxytocin neuron were labeled by fluorescent protein. In this case, green fluorescent protein. And we can find that there are two types of oxytocin neuron, one called mechanocellular and pulmonocellular, based on their cell size. And the topological analysis showed that different types of the oxytocin neuron actually expressed in the different brain regions. So they are all in the hypothalamus, but the different part of the hypothalamus. So when we stain of the oxytocin neuron and the vasopressin neuron, we surprisingly found that several oxytocin neuron also express the vasopressin neuron. So in here you can see the green part is the oxytocin neuron, and the red part is the vasopressin neuron. The sum of them are co-expressed with the oxytocin and the vasopressin neuron. So that also leads to us to the other direction. We also try to visualize the topological distribution of the vasopressin neuron. So we performed the in situ hybridization for the vasopressin, and we found that the oxytocin neuron and the vasopressin neuron, they are very close, so just adjacent. And in the different brain regions, we can find the cell size of the vasopressin neuron is different, so from very small to very big. So that also can code the mechanocellular cell and the pulmonocellular cell. So when we counterstain with the oxytocin, we can see the different part of the brain region. We also can find the co-expression situation. So here the yellow cell means that they all express oxytocin and vasopressin, and the red cell indicating that they only express vasopressin. So we account the population of these cells. So we found that almost 50% of the vasopressin neuron, they are also oxytocin neuron. So this is very unique and very rare because, when we moved to the mouse brain, and we found that they are close but dramatically different. So they are exactly separate. So in mouse, the oxytocin and the vasopressin neuron are separate. So we performed several mouse brain, and we only found one cell here,
they co-express in the oxytocin endovasopressin. So the other cell types like dopaminergic neuron, so we counter with pH, so that is the marker for dopaminergic cell. So we found that the oxytocin neuron are not dopaminergic neuron. So we make a summarize of these cell types. We found there these oxytocin neuron, they are also GABAergic neuron, glutamatergic neuron, cholinergic neuron, but some of them are also vasopressin neuron. They are not dopaminergic neuron and not serotonergic neurons. So that means in zebrafish, so these types of neuron, they are kind of multiple function, very different from the mammalian. So to describe and understand this cell more clearly, we decided to perform the whole cell patch clamping technology and the strategy to patch this neuron to see the electrophysiological features of these neurons. So we generate oxytocin neuron plus labeled fish. So the fish here, you can see that the oxytocin neuron are labeled by green fluorescent protein. Then we make the acute slides, make the brain cells still alive. Then we use the tiny pipette to patch this neuron and apply the current to record its electrophysiological response. So here you can see there are mainly two types of oxytocin neuron, cellular and mechanocellular cell. They are distributed in different parts of the brain. So here you can see the mechanocellular cell, they have a very high firing frequency, spontaneous firing. And compared to the mechanocellular cell, the cellular cell, they have a less firing frequency. And when we inject the current into this patched neuron, we found that the cellular property is very different. So over here, you can see when we inject the current, the free firing frequency dramatically increase in the mechanocellular cell, but increase slowly in the powercellular oxytocin neuron. So that indicates that in the mechanocellular cell, they have a hyperpolarization activated cyclic nucleotide channel. So you can see this sac shape of the waveform. In contrast, you can see the shoulder-like waveform in the cellular cell, indicating that there is A-type potassium channels in this. So powercellular cell and the mechanocellular cell, they exhibit very different cell features and function. So probably it is related to its projection of this neuron. So to do so, we use the same transgenic fish to visualize the projection of the oxytocin neuron. And we found that several brain regions, including the social behavior brain region, is highly innervated by these neurons. So oxytocin and vasopressin are already related to the social behavior, especially for the mating and the sexual behavior. So probably this is the reason. So after identifying the innervating the nucleus, so we try to trace the projection of this neuron, try to reconstruct the neural circuits of brain region innervated by the oxytocin neuron. So we developed a stereotaxis surgery of zebrafish. It is quite difficult because it is a tiny fish. So here you can see we use the muscle relaxing to immobilize the fish, but to keep them respiration, we need to insert a tube to the mouth and pass through this water, pass through the gill to keep them alive. Then we inject. So under this equipment, we can specifically inject the tracer to the specific region. Here, so here is the injection site. And by inject the retrograde and anterograde side, we can see the cell body. So retrogradely, we can see the cell body, or anterograde, we can see the projection. So we also apply the calcium imaging. So we generate once transgenic fish in which the neuron cell was labeled by GCAP. So the kind of a calcium sensor. So once the cytosolic calcium concentration increase, the GIP will increase the intensity of fluorescence. So here you can see the So when we stimulate the media raphe, so the other part of the cell are also activated. And here is the coronal section view. So there is a strong activation after we stimulate the media raphe. So here it is with the intensity of the calcium imaging. We can see the downstream nucleus of the ventral interpentacular nucleus, dorsal IPN, and the posterior part of the breathing central LA also increase the cell the activity. So by combining the neural tracing and the calcium imaging, we reconstruct the neural circuit. Okay, so which the oxytocin neuron innervates to this circuit. So for example, the habenula can be divided into two parts. They are projecting to the different parts of IPN. Then IPN reciprocally projects to the anterior posterior part of GC and the media raphe. And this neural circuit is related to social behavior, and especially for mating. So finally, we use the tissue clearance material to visualize the oxytocin neural projection, because compared to the mammalian, so mammalian brain, they have 7,600 oxytocin neuron number, and in fish, zebrafish and magnaca, only 300 to 400 neurons. So that allows us to give us an opportunity to visualize one by one. So we can visualize, trace the neuron projection one by one, and then we can patch this neuron, and we can distinguish the cell types of this neuron. So our hypothesis is that, so after the sensory input to the oxytocin neuron, and because in fish there are multiple functions, so they express of all kind of the neurotransmitter. Then after calculation, so different parts of the oxytocin neuron, they can respond to different parts, different stimulation of the environment coming from the environment. So in the end, I will thank all my lab members. So they generate all the data, and I would like to extend my appreciation to Dr. Huang Hongmo's fish physiology research family, especially for the support and the technical help for the Dr. Liao Bokai from Taiwan. And my collaborator from Institute of Molecular Biology at Academia Sinica and the Ken Center for Brain Science. And most importantly, my wonderful colleagues from National Taiwan University. So thank you for listening, and I'm very happy to take questions.
So I'm kind of curious about your cargo in terms of the ontogeny. Do they come from the same origin, separate?
Okay, thank you for your question. So basically, according to the previous speaker, Dr. Huang mentioned, the
differentiation of the oxytocin neuron from the stem cell. There are a set of the transcription factor to control the differentiation of this neuron. But now we don't know. So if the composition of the transcription factor can regulate the cell types of the two types of oxytocin neuron. But we believe so.
Originally the ventral portion as a ventral so I'm just kind of wondering together.
And also one thing I want to mention is that so when we inject the morpholino like orthopathia, so all of the oxytocin neuron were abolished or disappeared. So they probably share the common like a master regulator, but they also exhibit a specific because after the privation for the environment, so we do see the change of the different types of the oxytocin number. So under some situation, megacellular cell increase, but the other situation, cellular cell increase a lot.
Beautiful work. So the first slide you showed is the same as the slide shows. When you treat the patient,
yeah,
the GMP level,
yes,
right?
It increase. Yeah,
but also looks like the trajectory.
Yeah,
the connection, the big dot.
Yes. We are now doing that, but it is a very difficult work. So probably the resolution is not higher enough. So we try to use like TEM to quantify the data, the connection of the neurons.
Second question,
yes. Yeah, so because of the time limitation, so I skipped that part. So we found that the TRPV4 is probably the upstream molecule of oxytocin because when we knocking down the TRPV4, the oxytocin neuron also decrease. And in adults, the zebrafish, the TRPV4 are actually expressed in the oxytocin neuron itself. So probably because now we know that some oxytocin neuron they are also vasopressin neuron. So probably some TRPV4 also expressed in the vasopressin neuron. So we are working on that.
Thank you.
Thank you.
So you mentioned, for instance, the 50% of neurons.
Yes.
And vasopressin and also all the expression functional neurons. And do you see differences in this proportion, for instance, between
War on the cold temperature?
Yeah.
No, we haven't tried because so recent 1 year we found this phenomenon, and we are speculating that the increase of the oxytocin neuron under cold environment, they are probably the subpopulation of the neuron which also express in the vasopressin, because the oxytocin and vasopressin they all involved in the ion and osmoregulation. So probably only part the subdivision of this cell increase. The other because they are not, for example, not projecting to the pituitary, they project to the other brain region. So probably they do not increase so much. So that is our speculation.
And all the other functions, like I said, so did you check?
Thank you for your question. This is very good. So now we confirm that they are, for example, glutamatergic, GABAergic, and cholinergic. But now the next step is that we need to confirm for example, the receptor of this neurotransmitter actually exists in the downstream or the terminal of the exon terminal to prove that the function. That means oxytocin neuron, once they activated, they also release the acetylcholine or glutamate.
and the downstream nucleus is activated. So we have some data by stimulated oxytocin neurons. I mean the calcium imaging. So, but it is too preliminary, so I cannot show today.
So I wonder what is the involvement of stress hormone like cortisol? Because when you look at your temperature acclimation as well as the ionic challenge, you see similar changes. Suggests to me it's a common response to the stress. So what do you know about it?
Okay, thank you for your question. So, according to the previous lectures, so the cortisol also affect the activity of the vasopressin and so probably oxytocin neuron. So we are now doing the patch clamping. So we will apply several drugs. So, for example, we will apply the oxytocin itself because oxytocin neuron has been demonstrated to show the positive feedback of this neuron. So probably they more oxytocin will activate this neuron, and we will try to produce the acetylcholine or cortisol, these types of the molecule to check the activity of this neuron. So we believe we can find something.
Yes.
Somebody has a flashlight? I can borrow a flashlight. Oh, you have a flashlight? Okay, I can use her. Okay. I have a computer. So, yeah, I might need a borrow a flashlight. 先把我的装进去好了。因为我下午是一个泡所以。因为我刚刚那个老师是第几？他是明天的，明天的这个。对，对的。他的档案是这个，哦，这个是他可能要装这个才有办法。哦，才有办法。那个是那个老师要用的，对不对？对，但是这没办法装，所以他保持这个。确定看一下。有啊，可能有一些功能好像掉。所以这要怎么弄？所以刚才为什么大家有这么多？刚刚可能是他们用那个空者可能他们接收好像有点信号有点那么。哦，那我不知道空者要怎么用，现在是这个问题。然后就有两个，一个是可以用滑鼠的空者，还是你要用那个？用滑鼠的可能比较好。滑鼠的话，这个放大就。放回来这边。滑鼠的话放大，然后用这个。还是要用，这用哪个？我帮你用。 滑鼠的方向。
我还是要写啊。
OK， 好，那我要教你，就是放大之后嘛，右键。
哦，就还是平常的法。
对，一样的，没有变。对，你那个后面的PDF还是用鼠标，因为这打上他们没听不到。然后人家提问的，我刚按错，再重复一下。
好啊，很好啊，这没问题啊。好啊，好，没问题啊。好，哦，这
刚刚那个老师
哦，时运博，他明星的。
所以刚刚说他要装什么东西？
对，就他要换一个，
他要换一个。对，好，那个钟老师还给他了吗？还没送去。你的这个 USB。OK。我用了这种的，已经 copy 过了。好，谢谢啊。对，这个真的那个。好。我的这是已经 copy 过去了，我现在 USB。对，这说类似 USB。这是啊，那个改看日期。啊，对，我是这两个，这个和这个，一个是 PDF， 一个是PowerPoint。我先用这个看能不能用，能用我就好。我要改个名字对吧？这边我就给水永博，对，先看看啊。来了，好来这个 I open 就说。有时这个有时 Mac 和 PC 啊，你看这个就不是很重要，就是可以看，这目前来说还可以。就没大问题，唯一的啊，就是说它上面那个就挡住啊那个，所以我就说我如果看一下我的 PDF 就小一些 ，OK， 那这上面就不挡住。对，我来看，我把 PDF 放过来以后，对，把 PDF 也 copy 过来。我们可以试试看，在 Webex show 都会长什么样。对，你看啊 ，PDF 也是用 Webex 对吧 ？OK， 我用现在 PDF 的话是这个嘛？对，这可以选。这是 PDF 吗 ？Yeah， PDF。好，你可以放成 Play 对吧？啊，Play 是用哪一个？因为它是这是 PDF， 是 PDF， 但是我应该可以用这个。我知道，它一个 View 也是有一个 View。你来用了，我不知道怎么用。PDF 播放那个，它也可以用播放。哦，对，你看它这个，你看它小一些，上面就不会挡住。对，讲这个吗？对，这个我觉得就比较，对吧？就没事儿，对吧？这个啊。对，为啥你要改 PDF？ 对 PDF 我那个 pointer 是怎么用？就是看 PDF 可不可以用。PDF 的话，我直接用镜头就可以，我可以用镜头。哦，他可能用这个镜头。PDF 可能没办法用 pointer。对，所以我就用这个就可以了。OK， 好，这可以，对吧？可以。我是有影片。啊？你有 video 在里面？没有 video。对，这个没问题。对，这样的话上面就不挡住。对。好，行，那我就改个名字。好。我是有看，我姓石，我是明天早上第三个，所以是那个对方向来者是2，光改那个就好。这个哪个是？就是这个 PDF， 这个是 PDF 吗？这是 PDF， 所以，但是我这边前面帮你摆那个，你把它都是删掉的无所谓，后面这边让我们就知道了。对，我都不需要。对，所以我 到 P 2个就知道了。P 我之后把这个放在里面 ，OK 好，对吧？反正我现在不需要嘛，你就把也改成 SL，反正用这个嘛，对吧 ？OK， 万一如果到时候有问题，我们对，好，那我可以把这个抽出来，可以，然后我再打。好的，行了 ，OK， 好，老师，好，谢谢啊。我说什么啊？钟老师这个钟老师是等一下的，他是那个 PL2嘛。打开了。哦，
对，我觉得对，我觉得去外面喝的店，因为这个酱怎么办？
知道它里面的
太多台，
而且它没有因为里面的话可以重放，这个重
把这个冲
然后里面都没看的，
细菌群像。
不要我要干嘛？我干嘛？写长清楚，然后还没有干嘛？
没有张力。
那明天再做决定就是。
Okay, I think we'll get started. Good afternoon, everybody. I'm Tony Duan from University of Michigan. I'll be co-chair this session with Professor Qi Li. It's my pleasure to introduce the second plenary lecture. Professor Zhu Bangzhu. She's my hero. I told her, so I'm really looking forward to her talk. So without further ado, I want to give you all the 40 minutes. Please join me to welcome Professor Zhu Bangzhu.
Okay, thank you. Let me see if I can handle this. Okay, so today I'm going to talk about the control of the fish 6. Let me see. So, okay, so yes. So I'm currently located in the National Laboratory and Center. So I said this work was really done in the right? Okay. So about 6 development. So I usually compare zebrafish with 6 differentiation with mice. Yes. So with mice, because both mice and zebrafish are very popular animal models for medical and service. Yes. So both of them mature at about 2 to 3 months age. Yeah. Mouse is very commonly used. It has a very short gestation period, about 20 days or so. Start to, after giving birth, they reach sexual maturity about 2 months. Zebrafish develop much faster. Yeah, so zebrafish hatch for about 2 to 3 days after fertilization, compared to mice, that's about 20 days. So zebrafish develop much faster. But mouse 6 determination, mouse 6 is determined at about embryonic age 2 months. So at that time, we can distinguish this from moving. But zebrafish gonad do not differentiate until much later. So it has a very long delayed period. This period is very important because it interacts with the environment a lot. So zebrafish 6 is determined both by environmental factors and by genetic factors. So usually at about starting day 10 or so, we can start to see female 6 differentiation. And at about 1 month or so, we can start to see male sex differentiation. So we know that females always differentiate earlier than males. So this is very different from mice of the mammals. We will say that at this stage, with the gonad, you can look at it and you can see the difference between males and females. And fish, although it's a very popular model, receives a lot of studies recently, but its sex is actually quite difficult to study. The main reason is because the laboratory strains that they use, there are many laboratory strains, A, B, typical, and so on. And all these laboratory strains lack sex chromosomes. So, unlike mammals, we can check with the Y chromosome, DACA, you can check Y chromosome. But in fish, there is a sex chromosome to check. Yeah. And another thing is that the larval sex are indistinguishable. So if you look at the larvae in fish model, you cannot tell male from female. They all look identical. In fact, their sex can change all the time until some people say that after adulthood, they can still change their sex. So that makes it very difficult to tell whether this fish is a fish. But 10 years ago, John Foster would find that real wild type nadia that he got from the Ganges River has sex chromosomes. So they also determined for sure that zebrafish sex belongs to the fish system. That is the ZZ fish. The sex chromosome, when they are identical, they will develop to male. But if there's a ZW system, the W is for female. So this W locus is located at the tip of chromosome 4. And this region is missing in all the laboratory strains. That's why laboratory strains do not have sex chromosomes, although their sex is still developed regularly. Oh, it's not moving. So what should I do? It's not moving. Okay, so zebrafish, since it has the ZZ or ZW system, that means that it is the female dominant sex. So whenever there's an extra W chromosome, it becomes but it's not so simple. So because the W gene is required for ovulation, but a lot of times it's not sufficient. Okay, so a lot of ZW fish can still develop to males. Although without the W gene, ZZ fish always develop into male. So another difference is that we think germ cells are very important. So mammalian sex starts to differentiate from the Sertoli cells, the somatic cells. But zebrafish, it seems that germ cells are very important. Okay, so to understand this, so we first need to identify juvenile fish, identify the sex of juvenile fish. And so Eugene Pan did it using the traditional way. That is, we obtained the nadia, the real wild type nadia fish, and then identify their Y chromosome based on the PCR analysis using pair primer. So whenever there is a W chromosome, then W locus, then we can find PCR pair. But if it's a male, then we can see that this pair, this PCR product. But there are many sex reverse ZW males. So she mated ZW males with ZW females, then the offspring would have kind of different genotypes. That is ZW or W. the WW fish, when she made it with the GFP fish, PV1 GFP fish. So this fish will have germ cells. So then we will generate hybrid fish. This PV GFP fish is from a laboratory strain. So an Navia strain, maybe strain, the offspring will be contains W type. So this will, so you can see that because these are WW, so the W hybrid. When she tested 10 of them, all 10 of them had the W locus. So these we can consider as a future female. The ZZ fish, when it's mating with a laboratory strain that flourishes with the GMP germ cell, then the offspring we call Z hybrid. So this will eventually develop into females. So using this, then she can check. Yeah. So here she's doing cytohybridization. So doing in situ hybridization, here, so she's testing the W gonad. So using the marker SYCT3, this SYCT3 marker marks the synaptic complex. So it is an indication that meiosis. So here she can identify different types of germ cells. And using these different types of germ cells, so we can know that, for example, this type of germ cell, we call it gonocyte because it does not enter meiosis. But this enter meiosis at different stages, so different levels. So we have leukocyte, so on. Yeah, so she can count all of them, and she found that when she compared ZZ and CW gonads, she found that PGC primordial germ cells in the ZZ and CW gonads are all the same numbers, but the ones that differentiate into the gonocyte, then she finds that ZZ and ZW, then the ZW fish go into differentiate more than the ZZ. So this is consistent to our knowledge that the future females differentiate first. Yeah. And she checked at this stage. She checked the proportions of all germ cells, negative, negative, pre-negative. And she also found that the W gonad actually goes into zygote more than the Z gonad. So it also says that female gonads go into meiosis faster as detected by SYCT3. So here she examined the yeah. So if this is the case, then we decided to generate transcriptomes looking at fish of different ages, 10, 12, and 14 days, and check their transcriptomes. Yeah, so this is the transcriptomes comparing W and Z germ cells. And she found that the major genes that light up are the genes in the reproductive process, meiotic nuclear division, zonal synaptic. So these are all meiotic genes. So it says that at day 14, that's very early when the germ cells are barely differentiated. But if you look at their genes, all these genes for meiosis are already upregulated. So she picked 10 of these genes. Cytohybridization. So this is the cytohybridization. Yeah, VASA is the also co-state with VASA, so that the germ cells of the gonad can show up So when she compared W and Z chromosome, the gonads from W Z genotypes, she found that these genes, all these genes, express more strongly in the W hybrid gonads than in the Z gonads. It indicates that meiotic genes are also expressed more highly. So it's also another indication that just going to meiosis earlier. Yeah. To test this, she tested the SYCP3, the gene that I just told you that marks the meiosis. So she generated two different mutant alleles, mutations using CRISPR, and then checking these fish. And she found that these fish, when she checked their final sex in the adulthood, she found that in the wild type, there are a lot of types. In mutant, heterozygote also a lot of types. But two different lines of homozygous male fish, then they only develop somehow they just don't go into and she checked their germ cells using SYCP3. In the wild type gonad, she could see SYCP3 staining that protein, zygote cells. So this is normal staining. But in the mutant fish, homozygous knockout fish, then she could not find staining here. So that really indicates that that gene is disrupted. Another thing is that she can count the number of germ cells that go into, say, the protein diabetes. And she compared the number of diprotein cells, and she found that in wild type gonads, there are about, at this stage, about 5% of cells that will go into diprotein. But in the homozygous, none of them go into diprotein. It means that there is a meiotic arrest before diprotein stage. Yeah. So later on, because of this meiotic arrest, then all the fish do. So we make the conclusion that meiotic progression is required with fate. So without this meiotic progression, then fish change fate and become. So that was at the transcriptome we detected about day 14. So we also checked transcriptomes at earlier time. So this is at day 10, much earlier. And then at this time, then the pathways that came out were something like chromosome segregation, cell cycle, mitochondrial organization, then carboxylic acid biosynthetic processes. Yeah, this is a key match indicating that these genes express most highly and they tend go down. Yeah. So this means that like cell cycle, chromosome segregation, that cell proliferation. So we should check the cell proliferation marker by staining these gonads with PC. Yeah, marker synthesis. Yeah. So in the ZW gonad, she finds the PCMA staining here. And the ZZ gonad, she also found PCMA staining. So she counted the number of PCMA cells in the total gonad, and she found that the number of ZW gonad has higher than PCMA. ZW gonad this is higher. And she checked the germ cell number. And just to validate this, she just counted this germ cell number. In day 8 or so, germ cells are not proliferating. The numbers are about the same number of germ cells. So these are probably pre-meiotic. At day 10, they already go to some kind of proliferation, then we see higher number of cells. It also all indicates that WGS have increased the cell proliferation. So our first conclusion is that with such an early age, then cell proliferation, the increase of cell proliferation is an indication of their sexual differentiation. And this notion is consistent with the earlier experience. So earlier, many papers already said that in the biopotential group, we can somehow add more germ cells into the biopotential by injection, then these fish will to develop into we can lower germ cells like genetic mutation or adding dead end to kill the germ cells. So when the germ cells are reduced, then these fish will to male fish. So all these data all say that germ cell, the abundance of germ cells will determine. So it looks like germ cells are very important. So we want to study cells. But we know very little about germ cells. The only thing we know about germ cells is that they all start from germline stem cells. So germline stem cells has a marker too. So whenever that is positively to germline stem cells, and somehow they differentiate and they proliferate, and we see gametes, eggs or sperm. To get gametes, then it must go to but what's in between? We know that progenitor cells, these germ cells proliferate, so they go through multiple rounds of mitosis. But what kinds of progenitor cells are there? The only thing we know is that once they differentiate into prebiotic germ cells, then the mark will be that they will go. So we want to understand what's going on here. So we decided to do single-cell RNAs. Yeah. So Yang Jinxin took the 26-day fish, and these fish are marked. Their germ cell marked by GFP. So we dissected them, bunch of them, dissociate these cells, cell sorting to get GFP positive germ cells, and then perform single-cell RNAs. Then the data will be by circles, charge of the sequence. So we got the results, and we did not know what to do with the results until we met Professor Nico Zhao at the Institute of Statistics and his postdoc protocol. So they performed the bioinformatics analysis for us. So they used the Surya program to identify the cell clusters. So we got very few, actually around 800 wild-type germ cells. The reason why we got very few germ cells is because the procedure went through this procedure. It involves dissection, and dissociation, and cell sorting. This process has to be very fast, yeah, so that the cells are viable, are intact. So Jinxin could dissect about 20-30 fish at one time. So the number of cells that he got is limited. Yeah, so he got about 800 cells through multiple rounds of experiments. Yeah. But that was enough for our analysis. So we got about 40 clusters of cells. The connectivity analysis showing that these cells are connected, so they follow the trajectory. Okay, and the pseudotype analysis indicating that this is the beginning, so this is the base. We're going to start to the end of differentiation. So what are these cells? So these cells, by looking at their transcriptome markers, we can roughly differentiate them into three large groups. One group is the germline stem cell, that is W1. The second is progenitor, so W3, 5, 6. And W2 is in between, so we call it G to P transition. So germ-like stem cell to predict situation. And then the third group is meiotic cell. Meiotic cell group. Yeah. So the meiotic cell group then they express such markers as C1 or bug or produce that predictive. So since we already have some idea of meiosis, so I'm not going to talk about it. And nanos to represent so progenitor is something that's not known much, so we focus most of our efforts in progenitor. The marker for progenitor is FOXL2L that is expressed here from the. So we divided our progenitors into 3 different subtypes. So W3, 4, 5, 6. So they are now called early, committed, or late. They express different marker genes. For example, they are all nanos modifiers because nanos is a marker for germline stem cell. So they do not express any of the stem cell marker. And then they are FOXL3 is minus for FOXL2L, but positive for FOXL2L. So this marks the beginning of the progenitor. That's why it's called W3, the early progenitor, because it does not express FOXL2L. And the rest starts to express FOXL2L. The reason that I'm going to tell you later, we call it committed progenitor or progenitor C. And why do we separate the progenitor, committed progenitor, into 2? One is CS, committed progenitor S. Is because if you compare W4 and W5, their transcriptomes are very similar, except that W5 is very high for a lot of packaging synthesis proteins such as histone. So if you look at the histone marks here, you see that these are, of course, progenitors are strong. But the W5 here is especially strong in histone, S-phase marker. So if we perform a graph of marker for the S-phase, then the W5 will stand out. The other that will stand out is this one, W7. That is the pre-meiotic S-phase. W6 is positive for red 8A, so that's how it's differentiated. So these are different site hybridization data showing how to differentiate cells. For example, so the red cells are FOXL2, that's different. The green cells nanostructures. So that's how we separate these markers. So our summary for now is that we can separate these two cells into 3 categories: germline stem cell, progenitor, and meiotic. And for the progenitor cells, we separate them into early progenitor, committed progenitor, and late progenitor. We also show that these progenitors, early committed progenitors, so FOXL2L is expressed on the committed progenitor and pay special attention to it. The reason why we pay special attention to it is because we already know that transcription factor FOXL2 is a very strong factor, a transcription factor. So in mammals, in mice, we already know that there is an antagonism when undifferentiated gonad try to differentiate there is an antagonism between the systems. So FOXL2 is one of them. FOXL2 will promote the differentiation and repress the testis differentiation. ISOCCE is the other one that is strong for testis. So since we're looking at female factor, so we want to look at the fish FOXL2. And even fish has 3 FOXL genes, FOXL2 genes, 2A, 2B, 2L means like. Okay. And 2A and 2B are expressed in the somatic cells. So very similar to the baby FOXL2 cell expressed in the somatic cells. So 2L is expressed in the germ cells as you can see. Yeah, although they're not of sex reversal, so we know that 2AB is an important variant as well. But since we pay special attention to the system, you will see that it will show that it's germ cell protection. So this is the FOXL2 expression. So you can see that what site hybridization showed that it's we call it competitive project. So it's very different from so they never overlap with the mice. So, yeah. And then if you look at the expression, it's expressed the 0.56, this kind of cell cluster. So we know that germline stem cells still once they go into differentiation transfer, if they express then they become competitive projector. That means they go into competitive for then they will start to express reactive. So Wang Yanwei started to examine this FOXL2 expression. So in the adults, you can see this TL is laboratory strain, so it's expressed only. But if you look at Nadia strain, where we can differentiate, you can find that it is expressed also that they go there. Yeah, so one is the side but they all show expression before sexual differentiation. Upon sexual differentiation, the expression started then has certain things. But so it becomes sex bias, sex sensitive period. So Yanwei generated mutants, deficient. First phenotype that she saw was the homozygous mutants. So there's a sex reversal. This is complete sex reversal because when she checked it, the fertilization rate, she found that wild type of homozygous knockouts. So this is a complete big change from to cells. Unlike their germ cells cannot progress, but this is and then Jinxin detected their germ cells at about 28 days. The wild type, he checked two different markers. These are spermatin marker. He detected the expression of these spermatogenic markers in the gonads. They are missing. That means that these homozygous knockout fish actually develop testes precocious. So we know that FOXL2 controls the so the mutants become and mutant germ cells fail to enter. So what does mutation? How does it appear to be? So Jinxin performed single cell RNA sequence mutant cells. Yeah, so he got a lot of transcriptomes. He compared the transcriptomes of mutant cells with the wild type cells. So whenever there's a match, then he put them together. So he found that most of the mutant cells can match the wild type W1, W2. So these cells have W1 cells can match. But starting from W6 on, there's match. That means the germ cells somehow stop rest at this stage. Yeah. So he did in situ hybridization to check two different markers. REC8A, which is the marker late progenitor, as licensed marker. So he could find the marker expression control, but the knockout nothing. It also says that the mutant don't stop, the germ cell stop at the competitive progenitor, cannot progress further. So our small conclusion is that somehow when we knock out FOXL2, they cannot progress. So they go into. So if this is the case, then we check these cells. So these mutant cells, since they match the wild type cell in the W1, W2, so we wanted to know how close their gene expression pattern. So then we check their differential gene expression. So germline stem cell 1, 2, and progenitors early committed. Of course, there's nothing starting from the day progenitor. But in these regions, you will find that the strongest differential expression is the committed. So in the middle, the color is thinner. Yeah. But going back to the GSC, then it's stronger. Yeah. So this is just looking at the individual gene. So then we took many of these. For example, for ID1, so in this wild plot, you will find that the wild type is expressed very low, the GSC part. Nanos2 is expressed also only in the GSC, but not in the later stage. But in the mutant, then it's very strong in the committed progenitor. It's much higher in the GSC part. And for DMRT1, this is a male marker. Then you will find that it is expressed strongly throughout. Yeah, so it's very high. Goes to essential FOXL2 the gene itself, although the protein is its RNA is very strong in the germline stem cell. So it says that the gene expression, aberrant gene expression, is in the progenitor. It appears that it's higher to these markers like ID1. Then there are also differences in but so there are differential gene expression, especially in the competitive progenitor. So competitive progenitor is opposing. And this is ID1 and Nanos2 are the two genes that are strong, that express very strongly in the mutant progenitor, mutant germline stem cell. Examine these cells more closely. So this is the wild type cells. Check Nanos2 is red and FOXL2 is green. You will find red and green cells. But in the mutant, some of them overlap, so you see some of the mutant cells. And this is in cipher hybridization. Red and green, but they are in the same cell. This is for ID1, it's the same thing. But there is very little ID1 expression. The wild type cells, they do not overlap with the mutant FOXL2 cell. But in the mutant, we see a lot of cells. So then we counted these cells. So the double positive cell we saw big increase in the FOXL2. So it says that double positive cells are apparently higher. So I think I just skipped to here. So the conclusion is that these cells go back to the germline stem cells. So that's what we saw. We saw this is very high. So they exit out of stemness very much. So we conclude that FOXL2 act early progenitor state, and then they commit progenitor. They suppress germline stem cell differentiation. They suppress persistence differentiation. So we now know the transcriptome of the genes starting from that, and especially the sex sensitive stage, FOXL2 is very important for progenitor differentiation. Persistence. So I stop here. Thank you. Any questions?
Any questions and comments?
To there is L2L. Okay, made up of all the other and the new so it transforms to one.
Right, yes. So Dr. Khanna was asking the difference between medaka and the fish. Because the medaka, the author of the FOXL2L is called FOXL3 medaka. And FOXL3 medaka had a slight different impact on the fish. In other words, that we just said that there's certain there. So it seems that in zebrafish, FOXL2L is very important in earlier stage for progenitor differentiation. So we know the progenitor differentiation cells arrested at progenitor stage. They don't go into late progenitor, nothing. So then the differentiation pathway just goes back to the dedifferentiation to germline stem cell. But medaka seems to you see stage that is so medaka is slightly different here.
Okay, then so we expression of what express story of L2L or FOXL2.
Yes, so the zebrafish FOXL has 3 members: FOXL 2A, 2B, and 2L. So you're asking what happens to the expression of FOXL 2A and 2B is what you're asking, right? Yes, because FOXL 2A and 2B are expressed in the somatic cells, they are absent from the different cells. So of course we can go back and check whether they suddenly reappear in the germ cells, but because in our transcriptome we did not see 2A and 2B database, but we can go back and check.
Thank you for the beautiful talk. I had a general question concerning this loss of the gene determination in laboratory strains. Did that happen repeatedly? And what do you think is the evolutionary pressure to maintain or lose the sex genetic determination? And final question with it, are there other genes lost together in this part of the chromosome?
Right, yes, so they are, all these are the question that everyone is asking. So the first one is about the, I'll talk about it later on. So the loss of that part, yes, we actually do not know. All the laboratory strains were generated 40 years ago. So major, there are 2 different labs. One is in China, the other is in Korea. So they did parthenogenesis and trying to maintain mutants. So all our laboratory strains actually encourage strains with paying attention to sex education. So we do not know how this is. So if we ask them, they say that it's out of selection in that area. So your first question is about evolution.
Yes, what could be the evolutionary pressure to use it or to maintain it?
Yeah, we know the six chromosomes tend to lose because they lack recombination. But there's so little studies about it, so we cannot say for sure. But in general, yes, six to reducing.
And the question was, is there any other gene region that has been lost? Directly involved in sex?
People do not know. Yeah, because this region, I think the so-called W has not been defined. I heard. So in the database, every version of the database
Thank you, Dr. Zhong, for the beautiful work. So what happened to the SYCP3 knockout female germ cell? Do they turn into the male germ cell? The reason why I'm asking is, at least in the memo, we know meiotic progression and the oogenesis are separate events. In the female, if you have STRA8, they fail to enter meiosis, but they still can progress to the oligo. That suggests to us the meiosis pathway or genesis pathway is separated. So I'm curious about your SYCP3 mutant. Are those stem cells dead or they linked to the male?
Right. So Dr. Yao is asking about the SYCP3 mutant germ cells. So this mutant germ cells are very different parts. These mutant germ cells die. Yeah, so we have done experiments to show that they died. And if we stop this by knocking out mutant tissue, then we can actually rescue them. So all these males are, typically males, but they don't have germ cells.
So in the rescue with P53, I would assume they retain female fate, the gonad.
That's right. Yes. So if you generate double mutants of SYCP3 and P53 mutant, these double mutants that we have the 1:1:6 ratio. So they go back to the female rate, although they're still the first.
Any other questions? Maybe I can ask one. So you mentioned one of the function of FOXL2L to drive the progenitor cell toward the female fate, right? We also mentioned on the last slide, they also contributed the de-differentiation of
Right.
that terms of, can you elaborate on those? Because I think you.
Right, yeah, because I was out of time, so I did not say much. Yeah,
that's important. Yes.
Right. Yes. So, these data show that the germ cells, the nanos2 is a germ cell marker. So you'll find that the markers are in the mutant. Yes, and then here, this is a different experiment where we generated transgenic fish expressing GFP in the FOXL2L mutants. And then here you will find that the nanos2 only the germ cell. The previous one is the double positive cell. This is just germ cells. It's also high. That means that you get more germ cells. So in other words, this also shows it's more germ cells. In other words, that they just go back to the germinized stem cell phase. So that's why we say that FOXL2L prevents the differentiation in germinized cells. So that's why I say that suppresses
That's great. Okay, well, thank you very much for a very exciting talk.
Thank you. Our next speaker, Dr. Pao Yao. Oh, Pao Yao Xiong. Sorry. Pao Yao Xiong.
from Auburn University, USA. His title will be Unique Pharmacology of Fish Neural Melanocortin Receptor and Origins of Melanocortin Receptor. If you are ready.
Okay, thank you. So I also want to start by thanking President Chang and Jung. Is that working now? So I also want to thank President Chang and Jung for giving me the opportunity to visit Taiwan the first time. It was so wonderful. I had to visit quite a few places. So today I'm going to talk about the neuromelanocortin receptors. So this melanocortin system consists of 4 hormones: alpha, beta, gamma, melanocyte stimulating hormone, as well as ACTH. They are derived from tissue-specific post-translational processing of a precursor hormone called POMC. So this is how POMC is processed, for example, in the anterior pituitary gland by pro-hormone convertase 1 into ACTH. In the hypothalamus and the skin, we have another enzyme called PC2. So these fragments are further processed into alpha, beta, and gamma MSH. We also produce another hormone called beta endorphin, but that works through opioid receptors, so it's not considered part of the melanocortin system. So these 4 hormones work through 5 of these melanocortin receptors. Again, they are all G protein coupled receptors. Today we did have quite a few talks about G protein coupled receptors. So they are numbered 1 to 5, but I'm just going to focus on these 3 and 4 because they are primarily expressed in the brain. I think Jeff Huang will talk later about MC2 receptor. So these 3 and 4 are primarily expressed in the brain. They regulate energy balance and some other functions. For example, recently we got a drug approved by FDA for treating postmenopausal women with low sexual desire because MC4 is directly involved in regulating sexual function too. This just highlighted too. I forgot to mention that we also have an endogenous antagonist for these receptors. Agouti is antagonist of MC1 receptor, and the AGRP, agouti-related protein, is antagonist of MC3 and 4 receptors. So this just highlighted where these neurons are. AGRP neurons are located more closer to the third ventricle, whereas POMC neurons, the hormone-producing neurons, are located more peripherally. So this slide highlights in that indeed this leptin-regulating melanocortin circuit is really critical in regulating our energy balance. So our fat cells produce leptin that can cross the blood-brain barrier, binds to its receptors. In the arcuate nucleus, we got 2 subsets of neurons express these receptors. One expresses our antagonist, one expresses our agonist. Right, POMC can be cleaved by these enzymes into alpha MSH that will activate MC3 and 4 receptors. So the molecules highlighted here in red, depicting that in humans, if you have mutation cause loss of function of any one of them, including leptin receptor, POMC, the enzyme, as well as our receptors that will cause early onset morbid obesity. So again, monogenic obesity. So these are some of the papers reporting the first mutations in leptin receptor as well as POMC and MC4 receptor. So this slide by Michael Schwartz really nicely summarizes what these 2 receptors, we call neuromelanocortin receptors, are doing. So MC4 is very easy to understand because when you activate MC4 receptor, you have decreased food intake, increased energy expenditure. So you have body weight decrease, right? So MC3R is very confused and difficult to understand because it does not change food intake, does not change energy expenditure. Rather, it affects feed efficiency and fat deposit. So when you activate the receptor, you can take in the same amount of energy, but you deposit much less fat. So again, in our production animals and our humans, we want to achieve totally different goals. In production animals, we want to increase our feed efficiency. In humans, you want to enjoy all the nice food, but do not gain any body weight. So again, this is the original knockout experiment showing that there is no food intake, but the fat cells, including both brown fat and the white fat cells, increase in size. So you have increased adiposity. So recently, Roger Kong at the University of Michigan also showed that indeed, perhaps MC3 receptor is very important in a process called regulating energy rheostasis, essentially the boundary of the energy balance. So he is saying that, okay, when you have MC3 or receptor loss of function, the boundary is much wider than when you have a normal functional MC3 receptor. So very early on, we studied some human mutations with this receptor. We indeed show that, for example, this is a mutation that first reported in Singapore, showing that, okay, the mutation has no effect on ligand binding, but cause a total loss of signaling. There's absolutely no signaling at all. So again, suggesting that MC3 receptor is also important in energy balance. So by now we've identified quite a number of naturally occurring mutations in humans in this receptor. With the MC4 receptor, the effect is really, really dramatic. This was the first knockout paper showing that when you knock out, these are the wild type mice, right? Each line is body weight of one mouse, okay, measured at different times. And then you can see this is the wild type, male and female. When you have homozygous knockout, the body weight is about twice as much as the wild type. The highlight here in the red is that they are heterozygous. So you have one functional allele, one allele, you still cannot maintain your energy balance. So again, we have a haploinsufficiency here. So one functional allele is not good enough. Again, these are just a picture. You don't need to do any statistics with these experiments, right? So again, by now, this is an older paper. We have about 250 mutations identified in humans. I highlight here a few in blue. These are the opposite of what you were expected. They have gain of function, but still identified in obese patients. These two red mutations, red polymorphisms, are associated with protection from obesity. So really good for if you have those SNPs. If we study the functional defect of all these naturally occurring mutations, we can classify them into different categories. Class 1 is that you cannot make protein. Usually these are nonsense of frame shift mutations. Class 2 are you can the proteins are translated, but they cannot fold normally. They cannot pass the quality control, so they can never escape the ER. We've got a very few mutations trapped in the Golgi. I didn't depict here. They are also class 2 mutations. Class 3 is that you can traffic to the cell surface,
but you cannot bind to the ligand. And then we have class 4, everything trafficking ligand binding is normal, but you cannot activate heterotropic G proteins. And then we also identified quite a few that can have all the normal pharmacology. So we don't know whether those mutations are indeed causing obesity or not. So if we put all these different classes of mutation, again, this is Roger Kong's paper from Michigan. He first reported a crystal structure of the MC4 receptor. If we put these different classes of mutations into the three-dimensional structure, we can see that indeed the class 3 and class 4 mutations are located where they are supposed to be. One is what was the extracellular part for binding to the ligand, and then the intracellular part for coupling to the G protein and activating the G protein. But very interestingly, the class 2 mutations can form a channel in the three-dimensional structure all the way from the extracellular to the intracellular part. And the class 5 mutations are more scattered throughout the receptor. So you will ask, okay, this is a comparative medium, you're talking about humans, we don't care. No, indeed, there are similar mutations have been found in fish too. So Manfred Shatter reported in swordtail fish, right? The mutations in this receptor confer growth advantage, so you can grow larger. The males, the largest male has the reproductive opportunity to mate with males. So if you're big, that's good for you because you get the opportunity to mate. This is a paper from Cliff Tabin showing that in the Mexican cave fish, right? So he caught fish from the lake and the fish from the cave. He showed that compared with the lake fish or the cave fish, again, we know that they have increased appetite, increased fat storage in the liver. That's what we expected, but also more resistant to starvation. But the most important for us is that he identified the cavefish has mutations in the MC4 receptor. He used our assay showed that indeed the mutant receptor has decreased basal activity, constitutive activity. I'll come to this term again in a few minutes. So mutant receptor constitutive activity is decreased and maximal signaling is also decreased. He didn't measure the ligand binding or receptor expression. So if we study the fish receptor, indeed we show that this is Zhao Yang's work. He now works at Shandong University. Still working on G protein coupled receptors, published much better than I do. So this was his work on both grass carp MC4 receptor and spotted scat MC4 receptor. So if we use the AGRP for the binding assay, you can see that again we always use human receptors control. So you get a similar displacement, right? But now if we look at the small molecules developed at the human receptors, you can see that okay, human receptor, you got displacement, but the fish receptor has no displacement. So here we are saying that, okay, these small molecules are orthosteric ligands at a human MC4 receptor, but in the two fish receptor, they are allosteric. That's why you cannot compete with the agonist binding to the ligand. We use iodinated NGP-MSH as a radioligand. So we show that okay, some of these small molecules and HRP are also antagonists at the MC4 receptor. So you shift the dose response curve to the right, right? But now let's look at let's say these are the biosignaling. So again, let's come back to the basal signaling, right? So these are the human wild type receptor basal signaling. Here I included 2 human mutation controls, but these are the spotted scat and grass carp. You can see the basal signaling is 8 or 10 times higher than the human wild type receptor. So again, later we've studied many more fish, MC4, they all have much higher basal activity. We don't know exactly what's the physiological meaning for the fish regulation of body weight, but it's a very consistent observation. So if we look at the wild type receptor, wild type human receptor, right? Several of these ligands are what's called inverse agonists. So by itself, it decreases the basal signaling. Okay, this Japanese compound MCL doesn't do that, but this is a compound developed by a company in Boston. This is Ipsen by French company, right? Again, they decrease basal signaling. Now let's look at spotted scat and grass carp. Again, spotted scat work was collaborating with Professor Li Guangli at Guangdong Ocean University. This was in collaboration with Professor Liang Shifang at Huazhong Agricultural University. So totally different results. Again, AGRP is still serving as an inverse agonist with basal signaling, but these small molecules, for example, in spotted scat doesn't do anything in the basal signaling, whereas in the grass carp, one of these serves as inverse agonist, but this still doesn't do. So not only they are different from human receptors, but even the 2 different fish, you have the same compound working differently in 2 different fish MC4 receptors. So if we measure kinase, again, John Sang already mentioned this, right? So the GPC are not only covalent activating classical G proteins, but can also activate kinase. The kinase does not necessarily have to go to the G protein, they can also go to beta resting and other pathways. So again, we can show that all these in both, let's see, all these grass carp and spotted scat, the antagonist is now working as agonist in the MAP kinase. We should earlier decrease basal signaling in cyclic AMP, right? But here it's a nice, very nice stimulation. So we call this biosignaling. So the one ligand at this receptor inhibits one signaling pathway, but activating another signaling pathway. But we show that indeed some of these small molecules doesn't do anything with cyclic MP, doesn't do anything with binding. So you may say, okay, they are not interacting with the receptor. But by measuring the kinase, we show that indeed they are binding to the receptor because they can activate the kinase signaling pathway. So they are allosteric bios ligands. So if we summarize, we show that fish receptors have very high constitutive activity, meaning that the signaling in the absence of any hormone, right? Again, we are comparing that with human receptor. Human receptor already has some basal activity. If you compare with empty vector transfected controls, that will be even higher, right? So we show that this endogenous antagonist, again, we use human antagonist. This is very interesting, has 5 disulfide bonds. So only one company in Japan can make this compound. If you want to pay an American company to make this compound for you to say, no, I cannot do it. So we always have to buy from this Japanese company with 5 disulfide, so we cannot make a fish compound, right? So we use the human one, but again in the cyclic AMP pathway serves as an inverse agonist, but in the MAP kinase pathway serves as an agonist. So again, this is what we call a power signaling. So I want to switch. How many more minutes I got? I want to switch just a few minutes talking about the origin of these melanocortin receptors. Indeed, in the 1970s, 80s, there were lots of papers, mostly using immunohistochemistry, showing that, in vertebrates, we can also make POMC-like molecules, so potentially making alpha message, right? But again, not in terms of really sequencing the gene or getting the protein, mostly immunochemistry, so you are not so sure how good those antibodies are. So this is a work by Rene G. He was an outstanding student in my lab. Already published 10 papers with me for a PhD, and got a few more working on. Now moved to Harvard Medical School for a postdoc. So we work on a few of these landslide species in the NCBI, the field suggesting that there may be MCR-like receptors. So what we did was we tagged with a mixed epitope, measure the surface expression. Again, some of these receptors are expressed nicely, right? Use human receptors control. A little bit decreased in the surface, right? So now let's look at a ligand binding nothing, right? So human receptor again, very nice binding, right? All four of these coating-like receptor doesn't do anything. What about our basal signaling? Again, you don't add any hormone. You can see our MC4 receptor, have human receptor has we set it as 1, right? And the other receptors can have 7 or 8-fold increase. This one doesn't increase them. But this one is even less than the human receptor. Okay, let's now look at the stimulation. Again, human receptor, we can get up to 50-fold. This is fold of basal. Okay, 50-fold of stimulation. This one doesn't do anything, so they are not responding to MSH stimulation either. So for the basal activity, if you do it more carefully, you transfect the different plasmid concentration. Again, you can see a nice dose-dependent increase in the basal cyclic AMP. This one doesn't have any basal cyclic AMP. Right? What about the antagonist? Again, very similar, doesn't respond anyway at all. So I think we also did MAP kinase, also no response. So this is suggesting that indeed these receptors are functional receptors with some with high constitutive activity, but they are not melanocortin receptors. Melanocortin receptors first appeared in lampreys with 2 receptors we call MC8 and MCB, but not in these landslides or these other codecs. Thank you so much.
Any question or comment? One or two at most. Any question? Yes.
Thank you for your talk. Yeah. So you compare human receptor properties and fish receptor properties. And my question is, among mammals, according to different types of regime and feeding, do you have differences?
You mean in fish receptor?
No, the MC4 receptor.
Yeah.
You mentioned the properties in human. And my question is, in other mammals, not in fish, are they the same properties, same constitutive, low constitutive activity, or do you have differences according to the type of regime?
In mammals, we studied giant panda, pig receptors, and they all have very similar basal activity. So, for example, panda essentially you are always eating right. So I think we also studied ruminants. Yeah, very similar basal activity, not dramatic.
Not related to the fact of eating all the day.
Yeah, no, yeah, no. Thank you very much. So the question was whether there was any difference in basal activity in the mammalian MC4 receptor depending on the feeding habit. And the answer is we haven't found any nice correlation yet.
Here.
Yeah.
Very nice talk.
Thank
So
you.
the idea that fish MCR4s have a higher basal level and there might be considered active is really interesting. But the data, how do you do the experiment? You transfect the fish phosphate into a human cell line, right? Okay. Did you look at the basal level of the protein and also stability? Because fish don't live in 37 days. Did they change?
Yeah, we did. So Dr. Duan's question was I did experiment 37 degrees at the fish. They got much lower temperature. So we can check if we transfer the receptors 28 degrees to see what is difference or not. In the fish receptor, because they have much higher basal expression usually is even lower. So if you correct for the expression level, the fold increase should be high. MCR yeah, so this is all species that have amount of numbers. It's not the same sequence. It's not the same 13 amino acids. So it doesn't matter whether the fish receptor, whatever, or human receptor, they have the same sequence. So use the same amino acid.
Now, our next speaker, additional physiological role of chicken upstream promoter subscription bacteria in homemade chicken.
Thank you.
At the time.
Thank you. Talk about our recent study. We have listened to quite a few stories about fish. Now let's pay attention to human. Okay. When you put the endometriosis in the Wikipedia, you search all the questions, all the answers appear on the video. And you can come up with this workout. The first thing you think about, the biggest one is pain, and then it's woman, and then it's disease. So this indicates that this is a very severe woman's disease that causes a lot of pain. And what is endometriosis? The prevalence rate of endometriosis is about 10 to 50% in the reproductive age group. That indicates 1 in 10 women suffer from this disease. There's a severe pain during menstruation, urination, and sexual intercourse. So it significantly reduces the quality of life of the woman. And also remember, if your girlfriend or your wife is not comfortable, you don't have good. So it also affects the women's families. So for women with endometriosis, about 50% will have infertility. So you can see that the lesions come outside of the pelvic cavity, the ovary, in the uterine body, or they can even attach and infiltrate to other organs such as colon, right? And this is a picture with endoscope. You can see the black one. Those are endometriosis lesions. Okay, so far, there's no cure for endometriosis. The medicine or the surgery paid to take care of this disease is just to relieve the symptom. So far, the treatments will be given to relieve the pain and slow down the growth of endometriotic tissues and try to improve fertility or prevent the disease from recurrence. So simply, it's just stop the pain. There's no way to cure endometriosis. Surgery and hormone treatment are two options to treat the woman with endometriosis. However, you can see there's a lot of side effects. For example, the surgery, there will be 50% recurrence within 5 years. And with the hormonal treatment, you can use a combined estrogen and progesterone, or just a progesterone only treatment. And you can see so many different kinds of side effects. Therefore, we want to ask the question, why is endometriosis so hard to cure? In the past 10 years, we summarized a study from our laboratory trying to show up the molecular mechanism, how endometriosis pathophysiology. And we can pay attention to the central part is the prostaglandin E2. And we come up that prostaglandin E2 can inhibit the phagocytosis of the macrophage. They can induce a lot of genes related to angiogenesis. They can also stimulate the expression of enzymes that can produce estrogen. They can even also directly induce the growth factor FGF9, so the lesion can keep growing even without estrogen supply. But what is the underlying mechanism caused the overproduction of prostaglandin E2, and why the pro-inflammatory cytokines can continue presence in the microenvironment remain unknown. So we work on this mechanism by targeting one transcription factor that's called chicken overactive upstream promoter transcription factor 2. Use COOP2 as a short. And I first linked COOP2 to the endometriosis when during the year of 2011, that I take a sabbatical leave, use them better College of Medicine in Professor Min Cai's lab, Cai Ming's lab. So his lab is known to work on the COOP2. And when I was there, I tried to think about how can we link together to work on that. And you can see that COOP2 can do so many different things that contribute a lot of human organs, biological process, and the cell fate. So when we look at the COOP2 in endometriosis, we found something very interesting. Today I'm going to tell you 3 different stories. How about COOP2 control the progression of endometriosis? So the first approach we took is that we want to examine the expression of COOP2 in normal endometrium and the lesion of the endometrium. And we found that it's very clear that the COOP2 expression level is significantly decreased in women's atopic lesion. So we can mimic this in mouse because mouse is an astral cycle. They don't have a maturation, so they don't develop endometriosis. But we can surgically introduce the lesion of the uterus into the mouse peritoneum or to create a lesion similar to that we saw in women's peritoneal cavity. And we can also see that the reduction of the COOP2 expression in the atopic lesion compared to the normal atopic lesion
tissues. So what causes the COOP2 downregulation in women with endometriosis? We think about this about the pro-inflammatory cytokines in the peritoneal fluid, and in women, because the oviduct that connects the peritoneal wall to the uterus, so there's a connection between the uterus and the pelvic cavity. So we propose that the pro-inflammatory cytokine in the peritoneal fluid may grow into the uterus and then inhibit the post expression. So we use peritoneal fluid from normal women and women with endometriosis and treat endometrial tumor cell, and we can see the ectopic peritoneal fluid can inhibit COOP2 expression. And then we test several different kinds of pro-inflammatory cytokines, and we found that TNF alpha, IL-1 beta, and TGF beta all can suppress the COOP2 expression. These three cytokines, they have different receptors, they have different signal transduction pathways, but they all suppress COOP2 expression. So we want to look at whether it's the common cause to inhibit the COOP2 expression. In this study, you can see when we inhibit DNA transcription, we can reverse the inhibition of the COOP2 expression. But if we block protein translation, we cannot reverse the suppression of COOP2. That indicates these three cytokines modulate COOP2 expression is at the RNA level. So the first candidate is microRNA. So we look at the COOP2 cerebral translate region through different kinds of species. We can find two microRNA 302 binding site. The MIR302A we can find here. And when we mutate the microRNA binding site, we can reverse the inhibitory effect by the cytokines. So that indicates these three different cytokines, they suppress COOP2 expression is mediated by upregulation of microRNA 302A. And indeed, we can see they can all induce the expression of MIR302A. And we quantify the expression of MIR302A and the COOP2 mRNA in normal and endometriosis women, and we can find this negative correlation between 032A and COOP2. So when we re-express the MIR302A, then we can reverse the inhibitory effect, but we will use an inhibitor that reverse the inhibition is reversed. And that also causes upregulation of COX2. So MIR302A inhibit COOP2, and COOP2 suppress COX2 expression. So when COOP2 is going down, the COX2 is going up. That causes the production of a lot of prostaglandins. And to prove that, we knock down COOP2 in cell, and we find that the overexpression of COX2. But in contrast, if we overexpress COOP2, we can suppress the IL-1 beta induced COX2 expression. So the cytokine, by inducing MIR302, and then suppress COOP2, and COOP2 suppress COX2. So minus means plus. So when we put on the inflammatory cytokine, we can induce a lot of COX2 expression. That causes a positive feed-forward loop to cause the pro-inflammation and overproduction of prostaglandin 2. So means post-donkey use the knockout mice. The COOP2 knockout is only in the stroma because this is a progesterone receptor-driven free expression. And the donor here we can see the COX2 expression is probably similar. But in the knockout mice, when we tissue the knockout endometrium into a wild type mice, we can see the over expression of CAX2 only in the stroma. The epithelial cell, because the COP2 is not up, so we also express a lot of CAX2. So this indicates that overexpression of prostaglandin by polypharmacy cytokine is mediated by COP2. So this is a summary slide to show that the polypharmacy cytokine here produce MEO302A that bind to the 3' end of COP2 and they suppress the COP2 expression. And because of the low level of COP2 expression, then the CAX2 expression is increased. That causes a lot of prostaglandin expression and cause one. That's one of the possibilities that cause the overexpression of the a lot of biological process. And during this study, we knocked out the COP2 in stroma cell, and we found out there's a lot of genes is regulated. In this heat map, just showing the angiogenic genes is increased when COP2 is knocked down. Especially here, the angiogenin and the VGFC. We were particularly interested in these two genes, and I will tell you a little bit about these two genes function in endometriosis. We can see that in human ectopic lesion, the angiogenin expression is increased, and this increase is at the mRNA level and also at the protein level. So when we knock down COP2, the angiogenin is induced. When we overexpress COP2, angiogenin gene is inhibited. And this inhibition is regulated by promoter activity. There are two COP2 binding sites and angiogenin promoter. When we intake the first binding site, we can reverse the suppression of angiogenin promoted by COP2. And the binding site 2 does not have any effect. So this proves that COP2 can bind to angiogenin to suppress its expression. So when COP2 is reduced, then the angiogenin expression. And overexpression of angiogenin causes the angiogenic effect. This is a in vitro tube formation case. We use human cell and treat it with conditioned media derived from wild type and angiogenin knockout tissue media and treated the human. We can see that if we use the control, then we can form very nice tube. But if we knock down the angiogenin, the conditioned media cannot stimulate angiogenesis. And we also use immunofluorescence staining to see that the angiogenin and the CD31 is the marker of microvessel, and they have line up very nicely here. And this is in human. Also, we can mimic that in the mouse ectopic lesions. So, COP2 knockdown can induce angiogenin and cause a very nice tube formation. But when we double knock down COP2 and angiogenin, we can reverse this effect. So, and the one of the stories that we found that the COP2 is also suppressed by hypoxic situation. So in the ectopic lesion, because there's not enough blood supply, so there's a high hypoxic stress, and this hypoxic stress suppresses COP2. So we demonstrate here that hypoxia treatment also induces angiogenesis because it can suppress the COP2 expression. But when we overexpress COP2, we can abolish hypoxia-induced tube formation. So in this part, we talk about like the study demonstrate that in the normal endometrium, there's a normoxic condition that human alpha is stabilized, and there's a lot of COP2. The COP2 suppress angiogenin, so there's not enough angiogenin to produce blood vessels. But in endometriosis, because the hypoxic stress and also the cytokine inhibition, HOOK2 level is decreased, and then it's did replace the angiogenic gene expression and cause a lot of angiogenic production and then angiogenesis. The other one is the VEGF. We also look at the VEGF-6 expression. But first, let me show you the CT picture. This is a 25 years old female. She has a very big ovarian endometrioma, as you can see here. It's a very big endometriotic lesion here. And the doctor also described that she has a enlarged lymph node just nearby the ovary. So that indicates that there's a chronic and severe inflammation, because if you have a region, that organ or the tissue has inflammatory cytokine, and then the nearby lymph node will be enlarged. So this indicates that this woman has a lot of inflammatory cytokine in the peritoneal cavity. So the VEGF-C we discovered here is mature form. That's a 30 kilodalton. It binds to VEGF receptor R2 and R3, and thus cause lymphoangiogenesis, the production of effective vessels. So we use the life one is a marker of effective vessel. We can see there's a very nice effective vessels, and also a lot of immune cells. The CD11C is a lineage of the multiple immune cells such as macrophage, lymphocyte, neutrophil, NK cell, etc. And VEGF-C concentration is high in women with endometriosis. And we can do the immune staining to show that the normal information there's a very low VEGF-C, but in the ectopic region we can see a lot of the VEGF-C. The two white arrows indicate there's a big macrophage also present in the region. We also collect women's peritoneal fluid and then measure the expression of the VEGF-C. We can see that there is concentrations increase with the severity of the disease. So there's a positive correlation of HOOK2 and no, I'm sorry, there's a negative correlation of the HOOK2 and the VEGF-C. And we can see that HOOK2 expression is high in normal and low in the woman with endometriosis. But this is in the ectopic region, and this is the ectopic region. And this knockdown of HOOK2 can also show the increase of VEGF-C. And this VEGF-C secrete to the cultural medium. So when there's a lot of pro-inflammatory cytokines, as we just demonstrated a few minutes ago, so we would like to see the decrease of HOOK2 is mediated by upregulation of the pro-inflammatory cytokines. And we can see here that the treatment with IL-1 beta or TNF alpha that will induce the expression of VEGF-C, but when we overexpress HOOK2, then we can suppress that. So this indicates that IL-1 beta suppress HOOK2 and then cause the upregulation of VEGF-C. Again, we do the in vitro tube formation assay, but this time we use the infected epithelial cell because this is the infected vessel formation. And we can demonstrate that treatment with the cytokine or knockdown of HOOK2, we can induce a very nice tube formation. But if we use the lenvatinib, the inhibitor of VEGF receptor, we can abolish this effect. So how can this VEGF-C travel to a far away, already established effective vessel of the lymph to recruit the progression of the effective vessel to the region? We think about that this VEGF-C is carried by extracellular vesicles. So we collect the medium and use narrow particle tracking analysis to demonstrate that women with endometriosis, the grade 1, has a higher amount of extracellular vesicles, and the size is about 50 to 150 meters. And then we correct the concentration of the cultural media and use TEM to examine this extracellular vesicle actually carry a lot of VGFC. You can see here the dense dark particle that indicate that VGFC is carried by the extracellular vesicles. So when we characterize that using EV and EV-free competition media, we can demonstrate VGFC is in the EV. So I will skip this one. This is to just demonstrate that the EV can be uptaken by the effective vessel, and then the EV from hepatic can stimulate the black cell migration, and especially when we knock down TYK2, the migration is very common. And then if we that's even higher than VGFC, and similarly, reaching the cell we studied cytokines, we can also see the migration of the effective vessel. So this is the mouse model. We can mimic that to create a lot of lesions, and as you can see here. But if we treat with the envatinib, you can see the lesion is very small. We correct all the lesions, and then we can see here that the treatment of the envatinib can significantly reduce the lesion size in the mouse. And also, we see the effective vessel is significantly reduced, but the blood vessel is not reduced by this because it inhibits lymphoangiogenesis, but not angiogenesis. Most importantly, as we can see in human, in the mouse, we really induce endometriosis. The infected, the lymph node nearby is enlarged. But if we treat with lenvatinib, then the lymph node is shrinking to the normal or even smaller than the mouse receives the endometriotic surgery. And with the reduction of the defective vessel, we can see here also the immune cell infiltrate to the lesion is reduced. So this demonstrates that mouse and human that the defective vessel is very important for the immune cell to infiltrate to the endometriotic lesion. Then, since the EV carry VGFC, we want to think if this can be used as a biomarker. So we withdrew blood from women, normal endometriosis women, we isolate the extracellular vesicles and measure the size and the fraction of the size of the EV, and we also measure the amount of VGFC carried by the EV. And then we quantify the amount of EV VGFC. You can see that in normal women, it's lower, endometriosis is higher. So we plot this altogether, 21 normal behave endometriosis women. We can see the survival curve is very good. The area under the curve is still 0.81, and this is very good in humans disease. So the model we see here is that the retrograde maturation create a polymerase cytokines that inhibit the expression of TYK2. Reduction of TYK2 causes the upregulation of VGFC, which induce the infiltration of infected vessels. And this infected vessel provide a high-speed railway for immune cells to infiltrate to the tissue, and that further cause the inflammatory microenvironment and produce a lot of inflammatory cytokines. And this is a vicious cycle that causes the continuously inflammatory microenvironment with endometriosis. Okay, so I hope that I convince you that The COOP2 here, they can do different things. One is to inhibit COX2 to produce more cyclin E2 that feed into our first part of the story. And then the reduce of COOP2 also cause up regulation of angiogenesis, cause angiogenesis. And more importantly, the reduce of COOP2 cause up regulation of AGFC and the lymphoangiogenesis. And this actually increase the infiltration of immune cell and produce more and more the cytokines to cause the prolonged and chronic inflammation within the microenvironment. So I would like to thank my collaborator, Dr. Nancy Wu and Dr. Sun. They provide me the medical expertise and also give me advice about the women's suffering. And also my students and my research assistants, and then the funding from NSTC and NHI. Thank you very much. Yes.
Thank you very much. Story. So I have examined 48 patients. One of the slides you have shown that. I was wondering what is the prevalence in detection of patient to is decreased. What's the prevalence? That's the first question. The second question is there's a threshold. You must be a certain level so detect the symptoms.
Okay, as you can see that the 48 patients put to level in the peripheral blood. Okay. Well, okay, we did not measure the COOP2 level in the serum because this we only collect serum in this cohort study. This one, the 48, we only respond. We did not check the tissue. If we want to examine the COOP2, we have to check the tissue. Right, but that is different cohort. Okay, in the tissue, we can consistently see the reduction of the COOP2 in every single lesion. Okay, in the ectopic lesion, when you compare to the ectopic, the highest is the normal luminous endometrium. And then the median is the endometrium of woman with endometriosis. And the lowest one is the ectopic lesion of endometriosis. So if we compare the ectopic and ectopic, all the ectopic are lower than ectopic. And if we compare that to normal, it's very low. Okay, thank you.
Thank you very much for that. I have a similar question as the previous ones. You clearly showed the COOP2 reductions. I'm wondering whether this is the major cause. Can you actually recapturate this by knocking down the COOP2 and see what you actually develop in the measurement?
Okay, thank you. To answer your question, I think no, it's not the determining factor for developing endometriosis, but it's a critical factor to promote the progression of endometriosis. Keep it consistent. And to your question, the first question, the cutoff, and VGF, we use 950 picogram per minute.
Okay, just let's say
okay, thank you.
This concludes this session. And I'll go to the dance. There will be a break. Quick break outside. 15 minutes. Okay. Now. 3:30. Okay. Okay.
before our
So again, what? Yeah, yeah, yeah. Kind of. That's a really yeah, yeah. So again,
是准备现在讲还是把视频？先试试现在讲好不好？现在放视频啊？对，现在讲完就是你了。现在他要放视频，所以说你无所谓啊。我叫你现在也是那个现在讲。啊，对对对，那等他完了以后就是你了。现在现在是对，然后那个3:30开始，先放他的那个啊，对，对，好不好？
我说这个现在有声吗？
那就这样吧，好吧？对。啊。上来吧，你现在加入可以啊。
然后我 share 一下，share 一下。
Hello, everyone. So the next session will be for online talk from mainland China. The first one is in Zhang, research fellow at the Institute of Hydrobiology, CAS. So we're gonna use video.
Organization of this conference. My name is Zhang Ying from the Institute of Hydrobiology, Chinese Academy of Sciences. It's my honor to give the talk here. The topic today for my talk is sex and infertility control in C. primate through genetic manipulation. My talk will focus on the studies on the steroid hormone for sex and infertility control in sea primate fish. First, I will give a brief introduction about the background of the synthesized pathway and the main functions of the steroid hormones. As you can see, the main sex steroid hormones are progesterone, testosterone, and estrogen. Progesterone is important for pregnancy supporting. Testosterone is important for promotion of male secondary sex characteristics, spermatogenesis, muscle growth, etc. And estrogen is important for egg and ovarian development. Our studies would focus on the CYP17A1, which is the key enzyme for the testosterone synthesis. Based on the synthesized pathway, without the CYP17A1, there would be no more testosterone and estrogen in the animals. In humans with the CYP17A1 mutations, they would exhibit adolescence presence with juvenile sexual characteristics, primary amenorrhea, and the pseudo commonly bright-eyed syndrome. In mice, before we began the studies, we only found the reports indicating that the CYP17A1 depletion would cause early embryonic lethal. In fish, Before our studies, only a report on a natural mutant of MEDACA named as SCL, which later found in the locus of CYP17A1, caused the loss of the male secondary sex characteristics. Some other related background works in zebrafish. The androgen receptor blockout caused more females with premature ovarian failure and now somatogenesis in mutant males. For the progesterone signaling, knockout of nuclear progesterone receptor would only cause infertility in females due to ovulation defects. We began to look up CYP17A1 in zebrafish at 2014. After we got the CYP17A1 fish, we found all glands of the homozygous mutant fish developed to testes with active spermatogenesis, but they all lost the male SSCS and the mating behaviors. This suggests that now essential roles for androgen signaling for testis development and spermatogenesis in zebrafish, which is very interesting. This actually brings us more questions. Loss of the androgen and the estrogen, then how this fish can still develop to functional testes with proper spermatogenesis, even with no male SSCS and mating behaviors. We found that defective male SSCS and the mating behaviors can be rescued by androgen administration in CYP17A1 zebrafish. The ovarian differentiation in CYP17A1 zebrafish can be restored with the androgen and the estrogen treatment. But when we restore the estrogen treatment, the ovarian differentiation process would be reversed in CYP17A1 zebrafish. We also found that the upregulated levels of pituitary gland autotrophins and the testicular factors in CYP17A1 fish. And this enhanced spermatogenesis in CYP17A1 zebrafish was seen at their 6 months post-fertilization state. However, if we introduce additional FSH beta depletion, the spermatogenesis in CYP17A1 zebrafish at 6 MPF state would back to the normal. From this paragraph of the steroid synthesizers, it is speculated and actually seen that the dramatic increased level of progesterone in plasma and testes in mutant fish. We also found the similar phenotypes in estrogen receptor and the CYP17A1 double knockout and the CYP17A1 double knockout fish. This further confirmed the non-essential function of androgen signal for testis development and spermatogenesis in zebrafish. Actually, we found a non-increased level of progesterone in androgen receptor knockout fish. But when we treat the androgen receptor knockout fish with exogenous DHP, it partially rescued the spermatogenesis in the mutant. This strongly suggests that the high levels of progesterone might be responsible for the proper spermatogenesis seen in the CYP17A1- fish. So we got the nuclear progesterone receptor mutant from Professor Liu Xiaotun's lab. When we made the CYP17A1 androgen receptor and the nuclear progesterone receptor triple knockout zebrafish, there is normal spermatogenesis in this triple knockout zebrafish. This indicates for the first time the augmentation of progestin signal can promote the testis development and spermatogenesis independently from androgen signals. Until now, we have some primary insights for the defects seen in the CYP17A1 deficient fish. The glandular differentiation into testes might be due to the high regulation of sperm cell development. The proper spermatogenesis is due to the enhanced progesterone signal. The missing of the SSCS and the mating behavior are due to the loss of the testosterone. It has been reported that the growth dimorphism in common carp leads the females grow faster than the males until their sex maturation. So we use our finding to make the new male and generate the all-female common carp population. This work has been begun at 2016. And indeed, we found that the CYP17A1 depletion caused a similar phenotype in common carp as those seen in zebrafish. During the severe outbreak of COVID-19, especially in Wuhan back in 2020, we generated our first batch of the all-female common carp in Guangdong province, which was the CYP17A1 plus-minus background. During the following two annual culture seasons, we actually found about 25% more body weight of the CYP 17A1 plus minus all female fish than the control male and female mixed population when they were raised in the same pond at their 1 year stage. We also send our fish for food safety assay and the pass reason. However, as our all female population fish are CYP 17A1 plus minus fish, we think we still need to develop a controllable sterility strategy for this gene-added fish. So we now have been developing the controllable sterility for this gene-added female carp, and we are making progress recently. We appreciate here for all the people working on this and our collaborators and the supporting agents. Thank you for your attention. Questions and the suggestions are welcome. Thank you.
Thank you. Then topic is open for questions. Topic open for questions. Before they ask questions. So if you think about if you overexpress progesterone by overexpressing progesterone, what do you think of the situation? Can you hear me?
Yes, that I'm not sure. I didn't overexpress.
Because you see the calculation of the CYP, you have infertility problem. But I guess if you overexpress progesterone, you might go into a female pathway, about the fertility issue. Did you hear what I'm talking about? I just said an opinion.
Thank you, Dr. Professor Yin. I have one question. So you think the growth induced of the stimulant enhance the growth of the all female? What is the reason? Is due to the monosex infertility and the reproduction energy transfer to the growth, or do you have some speculations?
For the first question, why would the female grow faster than the male?
Female grows faster than male?
Yes. The female grows faster than the male. Why is the mechanism of the reason? We are still working on it. We don't know why. But I don't think because we haven't got the infertile population yet. So I don't think it's the reproduction energy saving for this faster growing. Okay, thank you.
I mean,
My question is simple. For all female common carp, right? You get a 20-30% more body, more weight, right? If you measure the muscle, what about the muscle? The muscle get proportion more or less? So that's what matters, right, for the culture. The other, what about the food quality of all the population?
Thank you for the comment. We measure the body, the fish body are heterozygous female, a rationally proportionally they are the same as the wild type female. What I mean is they have same ratio of the muscle as the wild type female. And
for the conversation, a little bit more
protein is in the heterozygous female, but not significant. So what I mean is they are just normal as the wild type female for the slightly heterozygous female, but they grow a little bit even faster than the wild type female.
Yeah. Do you know why the CYT17 not on the top? It's infertile.
They are not infertile. They are still gameting. I mean, for the heterozygous. But for the milous mammals, they all become the glandle, all develop into the testes, but without mating behavior. So actually, they're naturally infertile. But we can connect the sperm and then fertilize with the wild type eggs. But let's say they are infertile because they don't think themselves as male, even though they produce sperm.
Any more questions? Now, thank you, Ethan. Thank you. Next. Just 在那边录。对，你自己共享就行。对，可以了，讲吧。OK， 下台。你声音稍微小一点，轻一点。
It's my pleasure to discuss this the indispensable role of the American one in sex determination and testis development in male television. And this one from Southwest University, Hongqing, China. It is well known that sex determination is the most fundamental biological issue, and one of the six is determined by multiple factors derived from somatic and junctures. Up to now, agriculture produces more than 80% of our aquatic food, and tilapia are the second largest group of species used for agriculture. China produces around 1.7 million tons of tilapia annually, which occupies account for maybe 25% or 1/4 of the world product of tilapia. So tilapia male grows faster than female. Understanding the molecular mechanism of sex determination is critical for sex control. This slide shows our visual for tilapia culture and reproduction. The greenhouse, the recirculating agricultural system, and indoor artificial propagation and acquisition of monosex prime with YY supermate and XX supermate. And this one shows the establishment of gene editing in tilapia. Up to now, we have constructed more than 100 gene mutation lines and achieved artificial control of rows, sex, and body count. So this slide shows generation of tilapia with different colors by gene editing. For example, we use pink, silvery, orange, golden,
and red. So also this one shows creation of transparent and fast-growing telomere by innovating. Now focus on our topic about the emerging one, sex. As we all know, many fishes display natural sexual function. This is related to the estrogen level. By adjusting the estrogen or control the estrogen level, we can also induce undifferentiated growth, for example, XY phase to the function of ovary and XX function of testes. This I call it the primary sexual function. And even we can change the sex of already differentiated ovary into functional male testes. So this is by long-term treatment with aromatase inhibitor, for example, Padrolol. These data clearly show estrogen is important for ovary differentiation and maintenance. Let's talk about androgen. By using 4 pairs of XY1 at different developmental stages, we sequence the transcript functions. And we found structure 2, and GSM MH -2. And what's more interesting is that we found a in XS1, we have almost all the cervical enzymes expressed in the bone. No other society. In contrast, in XY patient, we found none of this cervical enzyme expressed. So that means XX phase, even before sex differentiation, it starts to produce estrogen, not androgen. In XY phase, neither estrogen nor androgen. Consistent with this mutation of genes, estrogen synthesis results in sex reversal in XX phase. So this can be has been done in CYP11A1, CYP17A1, and CYP90A1A. All these mutants, the XX mutants, develop as functional male with testes. And even with double mutation of ESR2A and 2B, we also can get testes. So this is consistent with that estrogen is very important for sex differentiation. In contrast, mutation of genes for androgen synthesis results in no sex reversal in XY phase. For example, we take the 11A1, 17A1, 11C1, or 11HSP2 in XY phase. All of them are still there with testes development. So that means androgen is not important for sex determination. Then what's the role of androgen in sex? Then we perform simultaneous administration of E2 and TR and successfully induce secondary sex reversal of XY phase from 1 month to 3 months of from differentiated testes to different ovary. So these data clearly show androgen is critical for testis maintenance in XY phase. So then to explore the mechanism, we did luciferase or promoter analysis, or we measured the methylation level of CYP90A1A, and the results seems to suggest that androgen is very critical for maintenance inhibition because it can upregulation in a measured transferase and in XY phase. So the first inhibition is initiation inhibition by the main pathway, and the second one is by inhibition to suppress the later pathway. Again, by mutation, we identify later pathway as later pathway. For example, you take MHY, DMRT1, GSM, because male to female and the mutation of culture 2, culture 3, female to male. To date, the relationship among these genes is still unclear. Another one is expressed both in somatic cells and cancers in marriage. This is also true for selection. If you look at the immunofluorescence data and the single cell transcriptome data, you can see that the other one is present both in germ cell and somatic cell. GSF and MH are expressed only in the somatic cells. Consistently, mutation of DMRT1 leads to the reversal of XY finish and expression of segmenting MIA. As you can see here, the new XY mutants develop filter phases over and expression is expression of mutants. Culture 2. As we all know, culture 2 is implicated in one place. And I'm calling culture 3 to be culture 2, culture 3. We might have culture 2 and culture 3. If you look evolutionary narrative, where culture 3 exists. So culture 3 is only observed in species is yes, not in usury. It's yes. And we know culture 2 and culture 3 and addressing selections and respectively. And we our early assumptions culture 2 is not just and culture 3 is both expressed in junctions, not selections. This is also supported by algorithm. As you can see here, culture 3 expressed in junctions and culture 2 in selections. We also prove that culture 2 and DMRT1 and kinase whatever exists to determine disease. Like the difference, we see clearly culture 2 is S1 activating the antibody and the DMRT1 is suppressed. The co-regulation of SS1 to AYP and culture 2 are overlapping in the spectrums surrounding the junctions. So we also get overexpression of culture 2 and DMRT1 in excess fish. We found already to test this female to so this again supports suggests the function of operating units and DMRT1 units are really different. And when you mutation culture 2 causes 6 reversal XX male. This can be rescued by exogenous estrogen treatment vertically of mutants in the ovary. And culture 3, as you know, work as Professor Tanaka from Japan in 2015 and published the science culture 3 is the failure G067. Mutation of culture 3 results in somatogenesis in ovary. This is true in intelligent, so intellectual mutation of culture frame, we also get some of the differences in oral environment. What's more interesting is that we found expression of the other one for germs was activated in culture frame mutants. This is already visible and visible. You can see culture frame mutants if it's higher or clear inorganic expression in the cancers that you can see as presence. And the emerging mutation the rescue cancer section version caused by foster mutation. So in foster mutations, you have some mutations in the orange and some down mutation in the surgery. By promoting analysis and tissue sequence, we prove foster free that recognizes the presence in one transcription in a thousand those dependent manner and generally one that recognizes the presence of the free transcription in a thousand. This is neutral transcription of suppression. More recent, lead to a lot of doubt and critical differences. We found once the one is routine, the corner will not be investigated to optimal tests and mutation of failure has been in XY. For example, if the one is already, but this will not be investigated with a mutation of the failure capacity, say 19A or triple mutation. Or when the was lost. And it's why we have to mention about that in contrast. As long as everyone is 6 universal costs of taking on downstream. The blood mutation can be invested in excitation. For example, if this one is 17 mutation of MH1 over if you do or culture 2 or culture 3, you can rescue it to Texas, or you can rescue it by AI treatment. Aromatase imaging. The same thing is true for TFC. So that why we can get a functional test is this will be we found in these double mutants. Everyone was expressed, and in this one, everyone initiated testing appreciation. Based on these data, we make a molecular intelligence work to extend this combination therapy. We think antagonists between MK1 and factor 3 the germ cell determines the pigment cancers, and the antagonist between MK1 and factor 2 determines the pigment of somatic cells. And usually, the fate of the somatic cell, the fix is determined and the God 6 will be determined. Then also we found the sexual plasticity depends on the coding and distance of the MK1 factor 3. For example, in the right XY fish, which is true, you can get XY over. And in factor 3, use the XY fish. This is true. You never can get function over. And the treatment, you get the confident tests, but with your knowledge, you never get that is also the global was plasticity. So I want to See, usually I might not have so many same environment system. As I said earlier, focus rate observe the species is maybe originally loss of the user leads to 6 arm affected by accident. That's why we are otherwise, because we don't from our one story, if we first story, we all well, so there are also evidence highlights the importance of MRT1 in testing development of other species. But there is loss of MRT1 is widely. You can test this with non-junctions. You can DW individual, give you lockout, and this still will get off. In regular when the MRT1 locked down, even if you put mirror configuration, you still get off. It's one of the steps. If you only have one copy of one line, the other one is one. So the access space, you never change the six of this access space. And the never finish, you want to so taken together, these data clearly show the indispensable role of the MRT1 in sensitivity and the testis development embarrassment here. In the presence of the MRT1, even if you mutate MH1 or DSM, experiment into upregulating, you get ovary. But this ovary can be rescued by downward mutation of the female passing in culture 3, all treatment. In the absence of or if the MRI is mutated, then you can never rescue the female and mutation any female passing treatment mutation or by AI treatment. So I'm back to preventing MH1 and DSM was remarkably expressed in the somatic cells. Where the MH1 is expressed in both somatic cells and germ cells. Maybe that's why the MH1 is commonly used as the key neuroplastic gene in all work rates and even in some new work rates. So the take-home-message. When our data clearly assumes estrogen is critical for female sex determination and menstruation, where androgen is critical for male sex determinants. Once MRT1 mutates, the gonad cannot be rescued from functional testes. And once MRT1 exists, serious reversal costs of mutation of MH1 and DSM can be rescued. In fact, is the male passing gene indispensable for functional testes. So I would like to thank my lab members for their help and the government treatment methods amended by Professor Nisco event and Bruns and SFC and the Ministry of Science and Technology. Also, I would like to thank Professor for their help. Also, last but not least, I would thank organizer to give this talk. Thank you.
Okay, no question. Get very well understand the MRT hotel. Two of them they have directly inhibit each other. So in this case, any other upstream regulator configuration.
So, because we may give I think it's not usually you have different sexual practices, or immunologics, and also tutorial practice. So it's great. In this case, I say that why this meditation computer has
Can I have a question? I'm Zhang
Yeah. Okay, congratulations.
In the case of the CYP17A1 mutation, there is no more androgen and estrogen. But the might have in the earlier one, right? And now androgen anymore. But with DMRT1, they all differentiate to the testes, right? Yes. So what do you think? What's the target for the DMRT1 without the steroid hormone for keeping the glanders to the testis fate?
Yes. Still, the one sometimes it can come. That means the test is it's not necessary.
Yes. So what's the target for the MRT1?
That's what I also
Okay, congratulations for your great talk. And I would like to have one short question. When you treat the estradiol for even for the male with the DMRT1, then you get the functional female, right? Yes. So yeah, you get the ovary, and if you could get E2, yeah, and XY3, not a mutant, normal tilapia. You treat with estradiol, they got or functional female. Yes. So during the functional female, the estrogen still on the treatment, or you already finished the treatment.
You just treat them for very short period.
For how long?
1 or 2 weeks.
Oh, in a dark fish. Oh yeah. I'm asking the dark fish. Okay. Thank you. Okay. Oh, I got it. Sorry. Yeah.
Yeah.
Any more questions?
Excuse me.
Go ahead.
It's like you show that actually 等一下，就是讲的那个想法放大，
跟那个箭头放
不是，就是那边有个地方，你按了之后，它会有一个
是显示它嘛的，
就是不当时对，
不当时，
就是主要显示它的样子，就可以选。对，你看现在它就把它定住，所以它这个一定要显示。那另外一个显示的可能就是正在讲话的人。
那这样我是不是要从这样固定？但是我觉得是还没工作还是那个就是一直干。
这个我觉得可以。
It's all at the bottom because they never made it. And we check the whether they
所以我说，这线上老师在讲，我们就可以把它这个全部到线上。好。
Yes.
可以怎么调？到线上
These should maybe check with the students. That's not very well. But I feel
Thank you. We switch the chair.
Okay. So it's my great honor to introduce Professor Jianzhen Li from the Northwest Normal University, China, to present his story of a very interesting hormone. Okay, Professor Li, sorry. Thank you.
Okay, can you hear me?
Yes, we can hear you.
没有，是因为是没事。
没有说搞法，就他们的地区就
对，所以就你得一直
This is a hormone from germ cell, and this hormone has hormone activities. The most research interest is ovary oogenesis, always is the British has called. But during my study, I'm always curious why fish can produce for several reasons. You know, for each adult female British, they can produce hundreds of eggs each week. And as we all know, most agriculture can release lots of millions of eggs for fertilization. This species for the woman, this fish can produce thousands of eggs on rocks. Another example is memory loss. You can see from this figure, it's a chromosomatic index. This index is increased from 0.2% to 12%. We see 3 months, almost 6 months, almost 60 times increase. Only within half a year. So, fish require abundant energy to generate large number of eggs for reproduction. Why fish can produce so many eggs? And where does the energy come from? And also know how energy comes from food intake. In human, this question has already attracted much attention. We already identify a lot of hormones, for example, insulin, leptin, and other hormones. These important hormones are involved to regulate energy balance, food intake, reproduction, and many other processes. So the energy homeostasis is strictly regulated by hormones. So our hypothesis is some hormones are also involved in fish reproduction to regulate energy homeostasis to support the ovary development in fish. What is the underlying mechanism? It's possible a group in USA with the analyze disease, procedure very few compared to others. And after doing genomic sequencing, they found a gene called FB1. This gene has a mutation in C terminal, and luckily they found that in C terminal there is a furin calcite. Furin calcite means the effort for the furin can cut at this site. So they have attacked this site. So they cannot produce the peptide after this furin calcite. After analysis, they found this peptide can be secreted from the adipose tissue. So they named this hormone as asparagine. Asparagine can be secreted from the adipose tissue to the liver to stimulate glucose level. And subsequently, the second group also demonstrated another function of this hormone. This hormone can stimulate food intake, can increase appetite. So after knockouts in mice, the mice are thin compared to the white side. Then another group, they also found in human genome there are two different FBN genes. The previous one is FBN1, and they also identified FBN2 also has furin calcite at the same terminal, same as FBN1. And the FBN2 is only expressed in the calcium in human. So they named this hormone as presenting. They also found presenting as stimulate liver glucose level. So they published a paper. The title is presenting is a genetic hormone separated from human enzyme. But in human, it is very hard to study function. So we want to use another animal models like fish to study the function of these hormones. Compared to the human, the zebrafish also has three different FBN genes: FBN1, FBN2, and FBN3. And all FBN genes furin calcite is similar to the human, except zebrafish FBN2. In order to inform it, we perform mass analysis, and we identified a novel FBN2 alternative splicing form. This splicing form has furin calcite as the terminal. Although the location is a little bit different from the conventional ones. Compared to the other fish, most at a similar location. By analyzing the gene expression pattern, found this gene is only expressed in the testing and ovary. Using PCR, we found this gene is highly expressed in the ovary and testes. Then we performed in situ hybridization. We found FBN2 is highly expressed at early stage. In ovary and testes. This result also supported by the results of a single cell RNA-sequencing. All these results suggest that FBN2 mRNA is highly expressed in early stage cells in both ovary and testes. Then we generated the antibody for this peptides. We also detected the expression in the ovary and the testes. By using immunostaining, we found this peptide is only expressed in the early stage germ cells. This signal was further confirmed by the WASA co-localization. WASA is another gene for early stage germ cells, so we can see the co-localization at 35 and 90 DPF. In testing, we also found a similar result. The signal can only be detected at the early stage of spontaneous cells. We think this stage is a smaller body cells. And always the result is this peptide is highly expressed at early stage germ cells. Then we detected the signal at the embryogenesis. We found 7, 8, 9 was the fertilization. The signal is highly expressed in the primordial germ cell, and also some signal can be seen in the plant. Considering this high expression in early stage germ cells are being tested in cemeteries. So we name this peptide derived from cemetery cells into as dormancy. Dormancy, the full name is dormant germ cell processing. The Chinese name is Symposium. In order to analyze the function of this protein, we prepare the protein's components, and using this protein, we injected this protein into the enzyme separation. We found the signal glucose was increased after injection of this protein. Correspondingly, insulin was also increased due to the increase of serum glucose. So this protein, this hormone can stimulate serum glucose and insulin labeling secretion. Because glucose secretion mainly depends on the liver, so we tested the effect of on the glucose level in the liver. We found after injection of adenosine recombinant protein, glucose content in the liver was increased. The change of enzyme in gluconeogenesis expression was also increased. The protein level, for example, PCXB, the protein level was also increased. So this result demonstrates this hormone has stimulated glucose content in the liver. Furthermore, we also found that after injection of this protein, we can detect adenosine signal after 1 hour of injection. Some signal can still be retained in the liver, but not in other tissues such as hearts. And the use of adenosine can stimulate glucose level. The target organ is the liver, so adenosine stimulates glucose circulation by liver secretion. Because as protein, we mentioned that before, this as protein can stimulate protein type, so we investigate whether adenosine can also stimulate protein type in secretion. After injection of this protein, protein type can be increased after injection of this protein. If the antibody can block the effect of adenosine on protein type, after injection of adenosine several times, we can see body weight can be increased. Because this hormone is mainly secreted from the germ cell, then we tested the effect of adenosine on the germ cell development. After treatment of this hormone, we can see oocyte maturation can be stimulated. Panel A is a phenotypic change using PHP as positive control. And using adenosine antibody, this antibody can block the effect of adenosine on maturation. And after injection of adenosine, we can detect the increase of retardation expression in the liver. So this result demonstrates adenosine can induce oocyte maturation and retardation. In order to know the physiology role of adenosine in certification, we established gene knockout adenosine mutant certification using CRISPR design. After knockout of the gene globally, we found adenosine mutant is gradually died after 6 EPF. So we assume might be infected, but doing studies is still same as the white type. Green type was not affected in the mutant. But we noticed that interesting transmit has some problem in the mutant. And this phenotype can be rescued by overexpression of the gene. We also found tendencies also have defects. So this phenotype might be due to the defects in the intensity transmit and local intensity sequence mutants. Because all policy mutants are died, we cannot analyze the effects on other issues. So we established another lookout. This is a conditional lookout of gem cells of zebrafish. We established a method to do such conditional lookout using PV-1 remote and a crystal-Cas9 system. We successfully mutated bonusing sequence types in T and OP, but not in other tissues. And we also found the expression is decreased in the T and OP in this conditional accounts. And then we found out all are decreased in mutants in this conditional accounts. And you can see the size of the fish is dramatically decreased in this mutants. And for fertility and ovary development, you can see the ovary size, G size, and most ovary follicles are blocked at early stage. But in testes, we only found the size of the testes is decreased, but the historical gene analysis show that the different stage of the testes cells is similar to the white type. In order to confirm this phenotype, we established several different transgenic zebrafish. This is the family promoter transgenic fish. After the full expression of bonusing, you can see the fish size, body weight, body length, all are increased. For another transgenic fish, SC17, heat shock protein promoter. After heat shock, you can see food intake, blood glucose, and the expression level of testes enzymes all are increased. And because this hormone is only expressed secreted from gem cells, so we will express bonusing in gem cells by establishment of the SPV1 promoter drive to transgenic zebrafish. We found after overexpression of bonusing in gem cells can see fruits, meat type, ovary size, ovary weight, all are increased. This is further confirmed by several different other transgenic fish. And because bonusing is can be found in different fish species. So we use another fish species called lemon trout. We also see the purified types B2T, and the expression pattern is also similar to the zebrafish. This gene is highly expressed in the ovary and testes, and also highly expressed in the germ cell of early stages. And after injection of lemon trout, bonusing recombinant protein, you can see serum glucose was increased, also intact. It means the result of lemon trout is similar in zebrafish. So we summarize here Guanosine is a secreted derived from the NPM2 gene at the C-terminal, and guanosine is separated from the germ cells cytochromas, and this hormone can stimulate appetite and repose release at the liver, and also transmit intestine, and also regulate ovary development. This work has just published 2 weeks ago on Science Journal, and compared to the findings in human, because in human the name is empty. In fish, we name it as guanosine. Also, the original organs separated is different, but we think the function might be conserved from fish to human. In other fish, we demonstrated that guanosine can regulate energy homeostasis to support germ cell development because it is separated from the germ cell to stimulate food intake and glucose release to support ovary development. For human, we guess the function might be similar. This hormone secreted from the liver to stimulate female food intake and also glucose release to support fetus development. So we draw a picture. This image was just submitted to the ISOCCE journal, and this is a story about Yuan. Yuan is in Chinese mythology. Yuan is a creator of all mankind and human and the dynasties, and the Yuan whose gene is cleaved from the skin to support fetus development. Guanosine cleaved from the NPM2 gene to support fetus development is very conserved. And finally, thanks for this grants and students, and also collaboration. Professor Zhou in Stanford University, and also thanks for organizer. So my recent findings. That's all. Thank you very much.
Okay, so now it's open for questions.
It's a very nice talk. Problem is something. So the guanosine expressed in the latest stage of site, like 2, 3, 4, or just really like only early stage.
Can you repeat your question again?
The guanosine expressed in the latest stage of sites in zebra like stages 3, 4, 5, 10, beginning setting, we model exam. Look at the image, it's like more like a stage of the outside as well.
Yeah, you're right. Yeah. According to our results in the study, we can see high signal at stage 1, all sides. But I'm not sure 1A or 1B right now.
So in that case, and early stage development, if I should directly really with the work, but not really the latest stage. So what is the criteria?
It is possible. It is possible because it is separated from early stage. It might have some function at early stage, but right now we don't know.
So it's also possible to accept the guanosine also in the stage follicle cell.
This is a very good. Yeah, this is a very good question. Right now, the receptor for guanosine is still largely unknown. In the last year, someone identified a receptor for asparagine, but this receptor is only is just mediating the function for food intake, but not for other physiological functions for expressing hormone.
So just follow just now the questions. I actually noticed in your data you have one rescue experiment always experiencing at the CM report. So I assume this is that the human is going to express in a lot of tissues in the zebrafish. I think the data is that the overexpression of fluorescein would lead to significantly increase in size compared to comparison white fish. So I assume that the fluorescein will have a multiple function other than just stimulate hormone, but also better hormone. So basically, we increase the expression of fluorescein. So therefore, we can increase the size of the gene.
Yeah, you are right.
Okay, so yeah. Hi, Jensen. Professor Lee, would you still online? Okay. Okay. So it's my great honor to introduce Professor Su Tan Zhou from Fudan University to present a story on the female reproduction. Yeah. Okay, Professor Zhou, sorry. Thank you. Yes, we can hear you, but it's a little bit soft. Oh. Okay, much better. Okay. This is from the university. I want to first, I would like to thank Dr. Lu Yong for his invitation. My topic is about the hormone regulation of female sex. For a group, use the migratory loop is a model system. Our research focuses on a hormone is an specific hormone. Is named juvenile hormone. Juvenile hormone has dual functions. During larval stage, this hormone inhibits larval development. However, during the adult stage, this hormone regulates uptake. The red panel shows the action of this hormone through its nuclear receptor balance, MET, and so we hope we notice this protein in transmission of one and one. It is a primary early induced gene. This transmission factor can mediate both anti-metamorphic and metabolic actions of renal hormone. In addition to the procedure, 3 insects has been the next model studying the action of renal hormone in female reproduction, including regulatory law, also the other one is most different. Actually, the regulatory glucose has been a model since 1950 for the general hormone stability of actions. Because many insects have acknowledged over in the genetic after the DNA synthesized the antibody transport through the molecular and the cell, like the pigment cell there's a channel named Comparative. So VG has been passed through this circuit to reach the surface of the faucet. Eventually, uptake by the developing faucet. During the process of mutagenesis, there's two crises in the reproduction. The first, augmented stimulate massive is the first point. The medium that certain crises after the beach massively subside in the metabolic, how did transportation is over or pass through between the cortical cell and the body in the development process. Well, before under this crisis, I had interested to start the technique object to has the dual functions in the last stage adult stage, right? Everything is like expression. The adult stage is opposite regulation of the genes. So we found the tuner hormone can be induced RH1 phosphorylation. I already mentioned the RH1. Remember, early we found the GH induced RH1 phosphorylation occurs at the certain 154. This is triggered by PKCR. We further found phosphorylated RH1 and recruit C-terminal binding protein expression during the last stage so that to exert anti-metabolic functions. However, in the adult stage, the phosphorylated RH1 because activator named the binding protein for the inflammatory function. Now, this means the RH1 phosphorylation can recruit the pressure in the last stage and or the activator CDP in the adult stage eventually will have the pure function full stage. So we also found this phosphorylation and the inhibition of double metabolic is evolutionarily observed for the insect orders. From the very basic like the sulfur, like the local stress or higher like the alpha. Well, now I'm wondering to answer the question how does the GH stimulate the massive synthesize in insect cell body? So we notice the insect cell body as the polyloidization cell or then after adult emerging. The most cell are just stage of it. However, when the stage enters the mutagenesis, the cell could be 32. Now it's 32 copies. The stage there is a large increase of polymerization. So we found tuner hormone and receptor that have which induce expression of the DNA replication and protecting like the CDC or U5-1. Eventually, like get into cycle. So the cell and all the cells become polychloride. So gene is a lactose and to stop with the large scale, which is synthesized in the cell body. So this is what I mean. We also find the tuner hormone stimulate each type by inhibiting the macrophage. This macrophage, including like the 7 macrophage, RA,
with an aid of the cluster of vector RNA2 clusters expression. This RNA because it inhibits the gene expression, but the human hormone inhibits the expression of this RNA. Consequently, we enhance the target expression that's occurred one, not increase expression still be desensitized. Well, this example just shows how human hormone would induce expression or to inhibit the expression of target RNAs for action abroad. Actually, many insects that adult type need for reproduction. So we get the like the insulin signal pathway might be involved in the regulation of the reproduction. So the next question we try to ask is how does human hormone coordinate with insulin signal to stimulate reproduction? We have a very possible study. First, we found, like glucose has two insulin peptides, also insulin receptor. The limited or specified insulin 2 and the insulin receptor 2 highly expressed in adult metabolic and the finger itself. Well, we use the lookup to come to the price of IoT and the insulin receptor 2. We found, they have more efficiency of the production over development compared to like control IoT1 and insulin receptor 1. This means limited specific insulin peptide 2 and insulin receptor 2 play more important role in glucose regenesis. So next we find the force of the insulin and the human hormone are required for higher level of the excretion. This indicates either insulin alone or human hormone alone without the expression at a higher level. So we know we have to probably we can there is some of these we evolve so this is like the gene expression analysis, globalized analysis. We found the interesting and the belongs to CHLH and this other family named the USF are being stimulated factors. We found this one after this lockdown, this factor using the create expression of the DT and consequently the ovarian development is almost totally inhibited. There is no production of mature eggs. Very interestingly, we found the insulin induced USF expression at mRNA level at the finger. But the hormone will not induce expression, but can induce phosphorylation of the USF. For further study indicate both higher expression of USF and is also translational modification that phosphorylation both are required for USF function or reduce the expression. So we would like to investigate how phosphorylation was triggered by human hormone. We found there is RTP, PPA is involved. This involved in induce US phosphorylation at 3.184. We also found that the phosphorylate US only US phosphorylated and back to MET. This are called the MET is the receptor of the juvenile hormone. This binding partner hydrodimer is the IMA. This is belongs to the PHLH hostolin family. Well, in course, we force identify USI part for further in the USI and then MET form hydrodimers. These hydrodimers can upregulate the region expression, perform the different assay, we also perform the chain analysis, and there is a directory finding. Interesting is as the upstream of the immunogenic genes, there is a chain for the USF and the MET finding. However, the upstream of RH1 gene is the finding of MET with IMA. The additional or the no reject receptor content. We first interested all that and no access to interact with the life from the timer. In addition to MET with IMA, so by the very analysis, we found only is the continuing phase and at a higher GI actually at this stage, the genetic stage, the genetic pattern is much better than other stage. So at this stage, higher genetic pattern and the genetic studies, you have hydrodimerized with MET for functionally come back. This hydrodimers then expanded to the upstream of the esogenous gene. And in a summary of this part of the small work, we found that the proinsulin can induce the expression of the USF at primary level. Well, the tuner hormone induces the USF phosphorylation. So at a higher level, and also phosphorylate the USF protein and recruit or hydrodimerize with GH receptor MET. They forms the hydrodimer and binding to the VT upstream to directly upregulate the VT expression. As far as we know, the insect is the first binding to the juvenile hormone and use the dimer of its receptor with the USI. This dimer directly binding to the upstream of the VT gene to directly induce the VT gene expression. So after the VT is a antibody, the next question we ask, how VT will release from the antibody to the amylin? How VT transposes like the channel or species among the molecule cells. So also to teach the service of the process literally uptake by the preventive process. So we found the juvenile hormone can induce the pendency, actually the arrow indicates the pendency initiation by activating the ATP, the sodium oxide ATP, through its phosphorylation. This phosphorylation reaches the phosphorylation triggered by cascade, including the GPSA, RTP, PRC, and the PKC. Well, after initiation of the GNC stimulation through the ATP, we also found juvenile hormone can induce the physical development of the speed, not a lot more area. This is from not area cell are each other, but they can use simply protein, protein is made how sleep. This foundation is shaped by GPCR, GPC42, and ethical PDC is sleep. So for this mechanism, we have junior hormone has through the two different necessities to induce the position HPs, also for heart-rate phosphorylation to induce the genetic initiation and enlargement. Consequently, a lot of these of the outside the cell. With the of the cell, and adds to another signaling and out. The gene receptor phosphorylation. The phosphorylation in the VTR is activated. Partly the VTR could binding VG. The binding VG and VTR receptor is to uptake by the positive cells. So this work is published in PNAS. So for all this work up to today, we use glucose-based models to investigate all the possible specific harms we induce these large scale metabolic critical over uptake by the development process. This work is performed later by three young investigators in this case, I will practice the young investigator in my lab, also students, right students in my lab. I've done a lot of wonderful work. My work is supported by National Natural Science Foundation, the key project, also National Key Research Development Program. Thank you for all. Thanks again in this opportunity. Like to see your questions.
Okay, thank you very much. Thank you, Professor So, for the very nice talk. Anybody have any questions?
Thank you, very interesting talk. I have two questions. One is the earlier part of your talk. You mentioned that the insulin-like peptide and the receptor mediate the ovary size and also the egg production. But in some insects, the insulin-like peptide are dealing with the nutritional regulating nutrition in the females. So when you knock down if like peptide 2 and it is receptor, and you see some protein phosphorylation synthesis change. What's the US upstream factor? Is that directly related to the VG synthesis, or is something else in nutritional metabolism pathway also effect? Have you checked that? And
well, I think that is some of your questions because I don't know if some like the network doesn't work very well. Their question is about the newsletter and the hormone population. Sorry, I did not.
Yeah, that's the part you presented. But my question is, insulin receptor like pathway also regulating the nutritional metabolism and enough to provide the nutrients for VG production. But did you check or ever found something in the nutritional metabolism pathway? And you knock down this receptor and then you give like peptide.
This is a good question. Well, we have to check. There's a 2 years peptide and a 2 years receptor. But the evolutionary observed the insulin one and the insulin receptor one. We also check. Well, for USF, the major impact is USF who affects I mean, the insulin peptide 2 and the insulin receptor, not insulin receptor 1 and insulin 1.
Okay. The second question is when you talk about the GH-mediated patency formation for the origin to pass through the follicular cell and attach to the oocytes for uptake. And you mentioned that the phosphorylation is another factor which initiated the patency. So how after phosphorylation will that affect how the patency form? That means the follicle cell, how they shrink and then leave a space for the and we check that.
You mean this one?
Patency formation in the GH. Second part.
Second part. Here?
Yeah, here. So the question is, and GH is going to stimulate the phosphorylation a protein, and then you mentioned that the patency then form. But physically, how is follicles going to shrink through that protein's phosphorylation? How they going to shrink? Do you have any idea? We know in the South.
I will answer your question. I'm very sorry. It's not that. Your assumption wrong is the working out somehow. It's not clear. Your question is clear.
Okay. So Professor So and okay, so I have some questions here. So maybe you may already know the USDA, US Department of Agriculture just give approval for a company called Greenland Bioscience to give a trial on the new version of the pesticides, which is siRNA based pesticides. So I just want to ask the next question is, based on your study, we use siRNA against the USF as a pesticide to against disease.
We use the siRNA for this local. It works very well. So generally, at least 75% and 80%. Or you can say could reach 90%. Now we also get first recognize to look out. For right now, for most of the day, we have like average technology. For more intensive way, we still use the RNA, not necessarily.
So how do you deliver the
we just inject.
You just inject.
Okay. Yeah. Inject. So they will transport through the heavy to reach the cancer cell.
Okay. Great. Thank you. Yeah, I have no further questions. So anybody on the ground have any questions? Okay, so there's no more questions. Thank you very much. Thank you.
Thank you.
Okay. Yeah. Good. Probably devices. Yeah. Thank you very much for your very nice. Okay, so I think this afternoon we finish our section. Things everybody is paying. Okay, so for the people online, I think the session of this afternoon has finished. Thank you very much to attend this. Thank you.
Professor, can you stop your sharing?
Yes, yes, we will. So on the way, we will go to restaurant, but on the way, have a little bit about our marine research station. Yeah. Okay, and our bus here, you could put your stuff on the bus if you don't want to carry. If it is too heavy, you could put your box, everything on the bus. Our bus is waiting there, but we could walk, do a little bit exercise before a good dinner. Okay. Everyone invited. Carol, Chairman, you better come. Yeah, well,
关掉了。你要说完还是我帮你先关？哦，
看看要怎么个方法，因为直播要
可以先这个先
就
按一下它的
不是先结束全音。